Good afternoon, everyone, and good morning to the participants from UK, the panelists as well. Good morning, everyone. Okay, welcome everyone. So to the day 2. Chandrakant, can you please confirm if I'm audible?
Yes, sir, I can hear you.
Okay. So welcome all to all the panelists as well as the participants who are joining, who have already joined and are joining as we start the day 2. So this is FluxMAPS 2021 event. Day 2, the theme is 13C metabolic flux analysis in plant and microbial systems. So today we are having our keynote speaker, Professor Nicholas J. Kruger from the University of Oxford. And followed by that, we have 2 sessions. One is, both of them are on plant and microbial systems using 13C metabolic flux analysis. So we do have 2 breaks in between the opening session and the session 3 and session 4 for the whole symposium. So we'll wait for a while, maybe around 2:45. I will introduce the keynote speaker and then we start. And meanwhile, we will wait for all the participants to join from across the world. Okay, welcome everybody, all the panelists and the participants who joined from across the globe. So today, this is the day 2 program, which we are starting, the Indo-UK Symposium on Fluxomics of Microbes and Plant Systems. It's the FluxMAPS 2021. So the event is supported by SPARC and UKRI, UK India Education and Research Initiative, and jointly hosted by IIT Mandi and the University of Oxford. So I thank Professor George Ratcliffe and Professor Nick Kruger, who joined us today as well as yesterday, and has brought so much energy into the whole symposium. So I take this opportunity now to introduce the keynote speaker for the second, for the day 2. Yeah. So I take the pleasure of introducing Professor Nicholas J. Kruger from the University of Oxford, UK. He's the Director of Graduate Studies, and he did his PhD in plant metabolism at the University of Cambridge. And with immense experience in biochemistry for agricultural genetics, he has wide experience in industry before returning to the University of Oxford. And then he is an expert who contributed in carbohydrate metabolism and a pioneer in metabolic flux analysis of the central carbon metabolism, and mainly of the heterotrophic plant systems. Nick, I invite you to kindly give your keynote address, and I hope, Chandrakant, can you please share the rights to Nick so that he can share?
Okay, well, thank you very much, Shyam, for that introduction. I'm just going to try and decide which of these options I should share. I think probably that one.
Yes.
And I now can't see what I'm sharing. That's interesting. Bear with me just a moment whilst I try to.
You may stop sharing and resharing again. We can see the slides, but
you can see the slides. Okay.
Yeah.
I can't, but that's absolutely fine. Let me just. Right. I think I can see it now. Okay, so welcome to day 2 of this symposium. Yesterday, the focus of our attention was on flux balance analysis in plant systems, whereas today we're going to be concentrating on metabolic flux analysis in both plants and microbial systems. And I like to consider this as being a move from a consideration of the possible to the search for reality. So George Radcliffe persuasively discussed yesterday in his opening presentation the value of understanding fluxes through metabolic networks. And that idea was reiterated by many of the speakers throughout the subsequent sessions. Whilst the FBA, flux balance analysis, and associated approaches are valuable techniques for exploring the potential of metabolic networks, 13CMFA is the most informative approach for examining the actual fluxes that operate through metabolic networks in vivo. And this approach is well developed in microorganisms, particularly in the context of bioengineering in fermentation systems or other bioreactors. What I want to do today is to extend that notion by doing 3 things. First of all, by reviewing the principles of steady-state 13C metabolic flux analysis, or MFA. And then secondly, by considering the refinements of MFA that have to be employed for analysis of plant or plant microbe systems. And finally, I want to argue that FluxMAPS in themselves are not the endpoint of our analysis, but they're useful in providing an understanding of the function of metabolic networks. So before we begin, I'd like to just formally acknowledge and thank many researchers over an extended period of years who contributed to the development of my understanding of metabolic flux analysis, and the names of some of them are listed here. For some, their contribution has been by doing all the hard work, the experimental work, and providing the data that's forced me to consider my views. In other instances, it's just the questions they've asked have provided the stimulus or impetus for me to refine my ideas. And I think preeminent amongst this list of acknowledgements is George Radcliffe at the bottom, who's been my strong collaborator throughout the last decade or more, and has been responsible for asking those awkward questions or seeking clarification. Now, whilst all of these people have helped me develop my ideas, I should emphasize that the comments I'm going to make now today are my own personal comments, and none of these other people, other researchers, should be held responsible for any of the crazy ideas I'm going to suggest. So I'd like to begin by looking at the principles of 13C MFA. And MFA is based, and particularly steady-state MFA, is based on a relatively simple conceptual framework, which is that label from a precursor supplied to a metabolic network can redistribute into the intermediates and end products of that sequence of metabolic reactions. Under steady-state conditions, the influx and efflux rates are equal, and the labeling of the carbon atoms in the intermediates is going to depend on the relative sizes of the fluxes that produce those intermediates. So here in this example, we see that a metabolite can be converted to products through two alternative routes, one in which the positions of the carbon atoms remain unchanged, and in the other route where there is an assortment or reassortment of carbon atoms between the molecules. And this will lead to different combinations of origins of the carbon atoms in the products. The products then will be differentially labeled from the substrate depending on the relative rates of these two different processes. And that can be used in order to define the relative fluxes through two different sequences of reactions. Of course, the products of this reaction can in turn provide differentially labeled substrates for subsequent reactions in the network. And so the differences or the variation in the labeling patterns gets propagated throughout all of the intermediates and products of the network. So operationally, what this then involves to exploit this approach is firstly to conduct one or more labeling experiments. Typically when 13C labeled substrate is supplied to grow in cells, and this label is allowed to distribute between the metabolites within the network. The labeling patterns within the individual metabolites or end products can then be quantified using analytical techniques. And then those labeling patterns can be used to deduce the fluxes through the network through computational modeling. An important feature of this approach is that the measurement of isotopomers in this section here provides a very large number of constraints for the flux estimation. So typically in a 13C MFA experiment, we might get 100 or more isotopic measurements. And these are used to estimate the values of between maybe 10 to 30 independent fluxes through the central network of carbon metabolism. What this means is that the number of measurements greatly exceeds the number of estimated flux parameters. And this redundancy then leads to improved precision and improved confidence in the accuracy of the flux estimates that we can obtain. One other thing to note about this approach is that it requires skills in 3 different disciplines. We have to be good at metabolic physiology in order to ensure that the labeling is conducted under the conditions that we want the tissues or cells to be growing. Secondly, we require skills in analytical chemistry in order to determine the labeling patterns of the intermediates. And thirdly, we need to have familiarity with computational modeling in order to calculate the fluxes. Now, this approach that I've outlined, it has been developed and is now essentially a mature approach that is used routinely in metabolic engineering to quantify fluxes in model microorganisms. And many of the procedures and protocols for 13C MFA have been optimized and standardized for its application to microbial systems over the past decade. Nevertheless, despite its optimization, it's still worthwhile considering the factors that contribute to effective flux determination. And I think these can be considered under 5 headings. First is the experimental design or the labeling strategy. So this means identifying an appropriate combination of 13C labeled substrates and the types of measurements to employ. And the considerations that come into play here are, first of all, the cost of the isotope that is going to be supplied to the tissue. And secondly, the informativeness of the labeled form of the isotope in terms of how well is that going to isotope or labeled isotope going to be able to define fluxes. And that is going to depend on 3 things. First, the structure of the metabolic network. The second is the actual flux through the network, so the rates of the reactions. And thirdly, the nature of the metabolites or fragments of metabolites that can be analyzed to infer their labeling patterns. Now, although it's possible to use in silico simulations to identify optimum labeling strategies, you can see that this can only be done once we know, firstly, what it is that we can measure, and secondly, the fluxes through the system. But we're not going to know either of those until we've done the actual experiments. So we're faced with a dilemma, which is, in order to define the optimum labeling strategy, we need to know the solution before we begin. For this reason, researchers will typically use a pragmatic approach, which is simply to employ a combination of readily available labeled forms of the relevant respiratory substrates and hope that it gives them adequate coverage and informativeness of the fluxes through the pathway. The second consideration is the way in which the isotope labeling experiment is conducted. So for steady state MFA, it's important that, first of all, the system, the cells, are in a metabolic steady state, and that steady state is maintained throughout the feeding experiment. Secondly, the system has to be labeled to isotopic steady state. Typically, these feeding experiments will involve supplying the labeled substrate to microbial systems, maybe in chemostats, but in plant systems or non-fermentation bacterial systems, it would be done through batch culture. There's then a consideration of how many different types of labeled substrate should be supplied to the tissue. Now, generally, the greater the number of different labeled forms of the substrate, and the greater the number of replicate cultures, and the greater the number of metabolites analyzed, the better the precision you will obtain in the FluxMAPS. But generally, we find that it's rarely worthwhile analyzing more than perhaps triplicate samples of each of 3 different labeling strategies. And to use any more than that is simply to waste too much money or expend too much effort in their analysis. The third strand of thought then is the analysis of the labeling patterns of metabolites, or the metabolite isotopomers. And there are 2 general strategies for measuring the distribution of the 13C labeled within metabolites. The first of these is NMR spectroscopy, which in general terms will roughly provide information on the position of 13C carbon atoms within molecules. So it tells you which atoms within a molecule are labeled. The orthogonal approach is mass spectrometry, which in crude terms will tell you how many atoms are labeled within a molecule, but not necessarily give you much information about the position in which they are found. Now, in principle, both of these approaches can be used, and the results from the 2 can be combined. But for practical reasons, the mass spectrometry has become the more favored, the more popular technique, partly because of its greater sensitivity. And gas chromatography mass spectrometry is currently the most widely used technique for measuring the isotopomer label distribution within metabolites. There's then a question of which metabolites to measure. And amino acids derived from the hydrolysis proteins are the go-to metabolites to measure. Firstly, because they are easily extracted from the biomass of the labeled samples. And secondly, because analyzing the labeling pattern of amino acids gives us information on the labeling pattern of the precursors, which are a wide number of different metabolic intermediates throughout the network. So by quantifying the labeling pattern of amino acids, we are getting information on the labeling pattern of intermediates throughout the network of central carbon metabolism. But amino acids, or derived from protein hydrolysis, are not the only compounds that can be analyzed. Typically, glucose and other sugars derived from the hydrolysis of starch and other cellulose and other cell wall polysaccharides can be used. Sucrose and other soluble sugars can be analyzed, and a range of other compounds such as ribose derived from RNA, fatty acids, and other specialized metabolites such as hydroxybutyrate that might be derived from polyhydroxybutyrate in some microbial systems can all provide valuable information. And once we have that information, the next challenge is to employ it to estimate the fluxes through the network. Now, the problem we have here is one of a nonlinear regression problem that identifies a set of metabolic fluxes that will best reproduce the experimentally measured isotope labeling patterns within the constraints that are imposed by the rates of substrate consumption and product formation. And this is an inverse problem that needs to be solved by numerical iteration to find the flux distribution. So feeding labeled substrate to some cells will result in the redistribution of label and the production of a set of measurements of isotopic redistribution. And alongside that, we need to run in silico simulations in which we begin by guessing the fluxes through the metabolic network and then predict the redistribution of label, and so the labeling patterns in metabolites that would be obtained from that set of fluxes. We can then compare the measured data set with the simulated data set, and there will be some discrepancies, some differences. And on the basis of those differences, we can then modify the value of the theoretical flux, recalculate the simulated data set, and continue to refine the parameter values until we have minimized the difference between the experimentally determined data and the flux values through the network. Fortunately, there are a number of different software tools that have been developed to perform these calculations for us, and this is just an illustration of some of the software programs that are available. In fact, all 6 of these base their calculations on a conceptual framework of what are called elementary metabolite units, or EMUs. And the significance of this is that it reduces the number of equations that need to be solved, and therefore decreases the computation time. And there are pros and cons for many of these different software packages. Currently in Oxford, we favor Inca, which was developed by Jamie Young at Vanderbilt University in Tennessee. But I note that Tom Williams yesterday in his talk referred to 13C flux 2 for calculating fluxes through the network. And so any one of these software packages can produce a set of flux estimations. The final stage then is the statistical analysis of this model. First thing we need to do is to assess the goodness of fit of the model to the data. And this can be evaluated by considering the residuum, or the standardized sum of residuals, or SSR, which is essentially the disparity between the measured and predicted values. Assuming the model is correct, then the minimized SSR should fall within the critical chi-squared values for the number of degrees of freedom that we have in our model, which is the number of fitted isotopomer measurements minus the number of estimated independent fluxes and other parameters. So the flux estimates need to satisfy this chi-squared test. The second thing is to check for the distribution of errors, although this is not often commented on in MFA analysis. If the fit is valid, then the error-weighted residuals should be normally distributed with a mean value of 0 and a standard deviation of 1. And this can be assessed using a normal probability plot, which should generate a straight line. If there's a significant deviation in this distribution, that can indicate a problem with the fit. Specifically, a deviation from a straight line suggests inappropriate assessment of the measurement errors, or if there's a skewed distribution with a mean substantially different from 0, then it suggests a more severe problem with assignment of measurements or the structure of the model itself. Once we have a statistically acceptable fit, the data can then be used to calculate the confidence limits for the estimated fluxes. Now, in early days, these limits were approximated by linear statistical approaches near the optimum solution. But we now know that this can lead to gross misestimates due to the nonlinear relationship between fluxes and the isotope labeling patterns. So instead, the software that's currently available will generate accurate confidence intervals, and that can be obtained in two ways. The first is, as illustrated here, by evaluating the sensitivity of the minimized SSR to systematic variation in flux. So as we vary the value of flux through a particular reaction, so the SSR will increase, and we can define limits based on the shape of this curve. And this approach is often referred to as the parameter continuation method. The alternative is to perform Monte Carlo simulations in which multiple flux maps can be generated based on a population of data sets that are drawn randomly from the pools of isotope measurements that are defined by the observed values and their measurement errors. So typically, the approach would be to produce maybe 1,000 of 3,000 different simulated FluxMAPS, and then identify the confidence limits based on the values of fluxes across those simulated data sets. And one of the reasons that we prefer to use INCA is that all of these statistical analyses that I've referred to are found and are easily accessible within the INCA software package. So, as I've said previously, the application of these procedures to microbial systems, particularly those growing in well-defined fermentation systems, is now relatively routine, and it's led to what could be described as a pipeline for metabolic flux analysis in industrial microbial systems. Now, the great temptation then is to seek to simply apply this pipeline to plant or other microbial systems, non-model microbial systems, in its current form. But I would urge against indiscriminate application of this pipeline. There are a number of factors that we need to consider carefully in order to be able to adapt or modify this approach to make it suitable for incorporation of additional concerns and additional features that we find in plant metabolism. So I'd like to really, I think, highlight 5 different considerations that there are in implementing 13C MFA in higher plants and associated microorganisms. The first of these is ensuring that we define the correct metabolic model. This might seem entirely self-evident, but can be something that is worth careful consideration. We cannot always assume that the metabolic model used for plant metabolism we can't always assume that the metabolic network in plants will be identical to simply the major metabolic pathways that are identified in standard textbooks that you can remember from undergraduate lectures. I think there are further considerations here that we need to include. So, for example, we need to include we need to compensate. No, we need to consider unexpected processes that may occur. One example of this is that glycine can be produced from multiple sources in plant cells. Typically, in textbooks and in many metabolic models, the pathway of glycine synthesis is defined exclusively as being derived from serine. But glycine can also be generated during the turnover of threonine via threonine aldolase. And glycine can also be generated from glyoxylate via glyoxylate aminotransferases. And the significance of this is that it means that the labeling patterns that we see in glycine will be dependent not only on the labeling patterns in 3-phosphoglycerate, that is the precursor for serine, but can also be dependent upon the labeling patterns of oxaloacetate, from which threonine is derived, and also on the labeling patterns of isocitrate that can generate glyoxylate via isocitrate lyase, or the labeling patterns of Calvin cycle intermediates from which glyoxylate can be generated via the oxygenase reaction of Rubisco and subsequent glycolate oxidase activity. Now, all of these reactions are well known, but rarely, if ever, are these additional reactions included in our metabolic networks. And that's something that I think we need to consider carefully. Another example of where we may need to develop our understanding of the metabolic network comes from unexpected processes that may occur. So, for example, in a recent study of the photoautotrophic cyanobacterium Seneca cystis, researchers have identified a previously uncharacterized pathway of carbohydrate oxidation called the phosphodiesterase pathway. And this is quite separate from conventional pathways of glycolysis, pentose phosphate pathway, or the Calvin cycle. And in essence, it involves xylose 5-phosphate, one of the pentose phosphates, and results in the generation of a triose phosphate and acetyl phosphate. And flux through this pathway in Seneca cystis can be considerable. So under conditions where this cyanobacterium is growing on xylose and carbon dioxide, about a third of the carbon flux is occurring through the phosphodiesterase pathway. So we cannot assume that we know all of the pathways that are occurring within our system. And failure to include this route in the model certainly compromised the metabolic flux analysis and flux maps that they were able to generate, at least initially. The other problem that we can see with models is that we need to ensure that they adequately account for the labeling of metabolites. And I'm just going to give you one example of this, which is the assimilation of carbon dioxide in open systems. So cells growing on simple sugars as the sole respiratory substrate are going to depend on an anaplerotic reaction to provide precursors needed for growth. And that anaplerotic reaction is catalyzed by phosphenyl pyruvate carboxylase, or PEP carboxylase, and that requires a source of CO2. In the early flux models that were developed, the CO2 that was used by PEP carboxylase was CO2 that was generated by other decarboxylation reactions in the network. And in bacterial cultures growing in relatively enclosed sealed systems, that might have been entirely appropriate. But in plant cultures and any system that is highly aerated, there's a possibility that the CO2 that is incorporated into the system has been derived from atmospheric CO2 rather than metabolically generated carbon dioxide. And the significance of that is that the atmospheric CO2 will only have 13C at natural abundance, whereas any CO2 generated through the metabolic network may be labeled with 13C from the labeled substrates. So the alternative to this completely internalized system here would be to have one in which the CO2 that is used by the anaplerotic reactions is derived exclusively from atmospheric CO2. But this again might misrepresent the situation, because that atmospheric CO2 entering the cell is presumably going to mix with the internal labeled CO2, from which PEP carboxylase is then drawing its substrate. And a more appropriate formalization for this would be in this form, and the reactions that in the model that would describe this are here, in which the atmospheric CO2 comes in, mixes with metabolically generated CO2 to form an internal pool of CO2 that then can be used for anaplerotic reactions, and excess CO2 is then exported. So this pair of reactions is simply defining the gross CO2 input and the gross CO2 output. And CO2 output under these conditions would simply reflect the input of atmospheric CO2 plus the balance of CO2 generated by metabolism. However, perhaps a more convenient way to define this is as an exchange reaction between the atmosphere and the internal CO2 pool, and a second reaction that defines the net CO2 output from the model. So although this is a relatively trivial aspect, you can see how you need to think carefully about how it's going to be defined within the model for flux analysis. And indeed, similar considerations may apply to other inputs. So, for example, 1-carbon metabolism via tetrahydrofolate could be modeled in a similar way. The second issue is one of the fact that plant cells are uniquely compartmented. And we find that typically pathways of carbohydrate oxidation are duplicated in the cytosol and in the plastid. And under favorable, and this compartmentation needs to be reflected in the models used for flux analysis. And to do that, we need to define separate pools of metabolites and appropriate transporters between these two compartments. And under favorable circumstances, it's possible to resolve fluxes through equivalent reactions in these different subcellular compartments. Turns out that in order to do this, it relies upon being able to determine the labeling pattern in compounds that reflect or report on metabolites from different subcellular locations. So, for example, sucrose and starch, both of which are formed from hexose phosphates, are produced exclusively in either the cytosol, the sucrose, or the plastid, the starch. And so the labeling patterns in sucrose and starch will reflect the labeling patterns in the pools of hexose phosphates in the cytosol and plastid accordingly. And at least for an Arabidopsis cell suspension culture, when we compare the relative fractional abundance of the isotopomers of sucrose and starch, we can see that there is poor correlation between the pattern of labeling in starch and either the glucosyl moiety of sucrose or the fructosyl moiety of sucrose. what this suggests is that the labeling patterns of the subcellular pools of hexose phosphates from which these two compounds are made are different. Rather reassuringly, if we compare the labeling patterns in the glucosyl and fructosyl moieties of sucrose, there's a much stronger correlation. This provides clues that allows us to be able to discriminate subcellular fluxes. However, it has a corollary, which is that it means that the measurements of the labeling patterns of protein-derived amino acids in themselves are unlikely to provide sufficient information to resolve subcellular fluxes of carbohydrate oxidation in plants. And so it becomes important to get measurements of compounds other than the amino acids in order to resolve these subcellular fluxes. The third feature of plant systems is that they're frequently slow growing. And conventional steady state MFA is based on the assumption that the compounds that are being measured, in this case, the end products formed in biomass, have achieved isotopic steady state. But it might be impractical to wait until the biomass components have reached isotopic steady state, even if we can assume that the system can be maintained in a metabolic steady state for an extended period of time. So in systems in which the biomass or growth is relatively slow, an alternative approach is not to focus on the labeling patterns of the end products, but to measure the labeling patterns of the intracellular or metabolic intermediates themselves, because these intermediates typically have turnover rates in the order of seconds or minutes, rather than the macromolecules that turn over in times of hours or days. And so it might be possible to measure the labeling patterns of those metabolic intermediates at isotopic steady state after much shorter periods of labeling. If it's not possible to measure these intermediates with high precision, or they don't provide enough information to quantify the fluxes with high precision, then a further approach that can be developed is to extend the model by including the provision of pre-existing unlabeled material. And this is referred to in the microbial literature by the somewhat unhelpful term of a G value. And the G value is simply the fraction of a metabolite, typically an end product, that is produced during the tracer experiment and therefore is labeled. And so it follows that 1 minus G represents the fraction of the metabolite that was present before the tracer experiment and is thus labeled to natural abundance. And it's possible to include provision for pre-existing unlabeled material in the model simply by introducing additional reactions of this sort. A third feature of plants that needs to be considered is the analysis of heterogeneous systems containing 2 or more cell types. And this is something that George Radcliffe has alluded to yesterday in his introductory talk. And this idea applies both to differentiated multicellular plant tissues or organs, and to studies of plant microbe interactions, where we have two discrete, separate metabolic networks that are potentially interacting, but which are, if we're not careful, analyzed in combination. So my view on these systems is that essentially the same principles as described for subcellular flux resolution apply here with similar constraints. The models can include provisions for reactions to be duplicated in different compartments, but in order to be able to resolve them, then we need to obtain labeling information that relates to individual compartments within the system. One thing's for sure, which is that we cannot ignore this heterogeneity, and we cannot just close our eyes and assume that the average labeling patterns will generate a flux map that describes the average metabolism across the system. Again, I would stress these average flux maps are inaccurate, and that inaccuracy arises from the nonlinear relationship between the labeling patterns of the metabolites and fluxes through the reactions in the network. In many ways, this problem that we're considering here is similar to that is being considered by microbial biotechnologists who have begun to develop techniques for metabolic flux analysis in microbial communities. And there have been some strong claims made for the applicability of this approach. But in general, their approach is similar. The models that they employ include duplication of pathways and transport between the compartments. But it is still dependent on either data on the labeling patterns in metabolites in specific subsections of that network, or without that information, the fluxes that are resolved involve unrealistic assumptions of metabolism. And some of the assumptions that have been applied in these co-culture MFA studies include an assumption that the biomass composition of all the cell types will be identical, or that the different cell cultures, the different cell types are growing at the same rate and contain equivalent amounts equivalent pools of each of the metabolic intermediates. And none of these assumptions appear to me to be particularly realistic. The final feature of applying MFA to plant systems that I wish to consider is conditions under which the underpinning requirements for steady state MFA break down. And one of the most obvious examples of this is photoautotrophic metabolism. Again, as referred to by George in his talk yesterday, fixation or attempts to apply steady state MFA to photosynthesizing systems is doomed to failure because once the system achieves isotopic steady state, then all of the metabolites will become completely labeled. The rationale for this is that because 13C is being supplied as a single carbon substrate, there can be no asymmetry of labeling introduced into the system. The way to avoid this is, or to overcome this constraint, is to apply so-called non-stationary flux analysis. So rather than analyzing the labeling pattern when the system has reached isotopic steady state, we need to analyze the labeling patterns prior to achieving that steady state. So we need to follow the pattern of labeling as the label is introduced into the network and washes through the system. And that can be done. And there are facilities within the software that allow us to capture the data from these transient labeling time points, and from them infer the fluxes that are occurring in these networks, provided the system remains at a metabolic steady state. That brings me on to the final example of where breakdown of the underpinning requirements may occur. And that occurs in systems that fail to maintain a metabolic steady state to allow labeling either to isotopic steady state or transiently. And plants are dynamic growing systems, and in many ways, metabolic steady states are the exception rather than the rule. And I'm sure we all have our own image of changes that can occur in plant metabolism with time. I happen to have chosen one example here, which is the respiratory climacteric in ripening fruits of yellow monbeam. And in this example, you can see that changes in the rates of oxygen consumption and CO2 release, and the fluctuations in the respiratory quotient here reflect underlying fluctuations in both the rate and pattern of central carbon metabolism in these ripening fruit. And under these conditions, where metabolic flux is changing rapidly, it means that none of the approaches that I've yet discussed would be appropriate. What we really need to do is to develop an approach of what I might call dynamic MFA, in which we take into consideration both the changes in flux as well, changes in flux, as well as the changes in the labeling pattern. Unfortunately, there's currently no robust method for analyzing the kinetic labeling in systems where metabolism is changing. I suspect it would require accurate measurement of the metabolite pools as well as their extent of labeling. And the best we can achieve at the moment, I think, is to attempt to employ the non-stationary MFA over short periods over which metabolism is at a pseudo steady state, where it isn't changing very much relative to the rates of incorporation or redistribution of label through the network. So under these conditions, this is probably the best we can achieve. All of these considerations then will allow us to deduce flux maps for a central carbon metabolism in plant and associated systems. But those flux maps are not in themselves an endpoint. They're an important resource that can be used to seek to understand the network of metabolism. And I'd like to just briefly end by considering 3 different examples of the way in which the information from FluxMAPS can be employed to improve our understanding of metabolism. So the first of these involves testing specific proposals about the function of the network. And the example that I've chosen here is potassium limitation of metabolism. So based on a comparison of transcript profiles and metabolite profiles, researchers have suggested that one of the major direct consequences of potassium limitation in plant roots is to inhibit a potassium-dependent pyruvate kinase, which is responsible for the conversion of phosphenol pyruvate to pyruvate. And as a consequence of that, it restricts entry of carbon into the TCA cycle, promotes the accumulation of sugars, and restricts the production of amino acids due to a decrease in the availability of the organic acids that are going to act as the carbon skeletons for biosynthesis. Now, whilst the transcriptomic and metabolomic data are persuasive, they are only correlative, and we need direct measurements of flux to be able to test this hypothesis. So here, Sam Fletcher, in working with George and myself, has obtained flux maps for Arabidopsis cell cultures grown both in standard MS medium and in a medium that's deficient in potassium. And if you look into the details of these maps, you will see that there is decreased flux through pyruvate kinase in the absence of potassium. So we're confirming the expectation of the proposals that have been made. But this is only one of the changes that is occurring. There's a large number of additional compensatory changes that are not revealed by the initial proposal. Second way in which FluxMAPS can be exploited is to explore the sources of energy and reductant that are required for biosynthesis. So in a study,
Nick, sorry to interrupt. We have a couple of minutes.
Couple of minutes, that's fine, Shyam. Sorry, I've got carried away with myself.
No, so go ahead, please.
Okay, so in this study, in fact, it was Shyam, explored the effect of phosphate limitation on the provision and utilization of coenzymes. What he was able to show from analysis of these flux maps is that the NADPH required for biomass production halves between phosphate replete and phosphate deficient cultures, but that perhaps, and that the oxidative pentose phosphate pathway only provides about 50% of the reducing power needed for biomass production. The shortfall in NADPH production can be met by the activity of an NADP-dependent isocitrate dehydrogenase, but that depending on the phosphate status, the proportion of NADPH that is generated by the network that is required for biosynthesis drops from 90% in the phosphate replete cells to only 25% in the phosphate deficient cells. And there are similar observations that can be made about ATP production and utilization. ATP yields increase as phosphate availability decreases, but only a small proportion of that potential ATP yield is actually needed for biomass. So the general conclusion from this was that there is no simple relationship between the capacity to generate ATP and other coenzymes and the demand for these biosynthesis. And this clearly reflects variation in the demand of coenzymes for cellular maintenance processes. We can extend that in this third example, which is an exploratory analysis using euglena. And euglena can exploit a range of respiratory substrates. And here we're looking at the metabolism of ethanol, acetate, and pyruvate, each of which enters the metabolic network through a common intermediate, acetyl coenzyme A. But these different substrates have different yields of energy and reducing power. And this impacts on the coenzyme balances in the network. And that influences the proportion of the maximum carbon conversion efficiency that can be achieved by these networks. And this carbon conversion efficiency is calculated by comparing the proportion of carbon atoms in the substrate that eventually accumulate in biomass with the maximum proportion that could be incorporated into biomass based on flux balance analysis. The fact that, firstly, in no circumstance does this carbon conversion efficiency reach 100%. And secondly, the fact that it varies between substrates means that the flux through the metabolic network achieves a variable proportion of its maximum theoretical capability. And that illustrates the problem we have in inferring the actual performance of the network from the theoretical potential provided by FBA. So I apologize, I've overrun slightly, but I hope that today I've been able to persuade you of the following points. That first of all, steady state MFA is regarded as the gold standard for accurate and precise quantification of metabolic fluxes. It's a well-established protocol for defining flux in microbial cell cultures. And the approach can be refined and additional features incorporated to reflect the metabolism that we find in plant and microbial systems. And that finally, that the quantification of fluxes provides biological insight into the in vivo operation of the central metabolic network. So thank you very much for your attention. And if Shyam allows, I'm very happy to take a few questions.
Thank you, Nick. Of course, we do have a couple of minutes for questions. Yeah, let me read through this. Yeah, sorry about, I think my video. Yeah, now it is fine. So, yeah, this is from Kushboo. Thank you, Nick, for the excellent talk. Could I please ask about the biomass constraint equation in plant cell models? Eukaryotes with multiple subcellular compartments such as mitochondria. Should the biomass equation be a composite of both cytosolic and mitochondrial measurements? or can it be only cytosolic, the cumulative of all subcellular compartments? Sorry, it's too long question. Yeah, so it's any indications in relation to the biomass constraints equation, especially in the eukaryotic models?
Okay, right. I'll tell you what we do, and then what we perhaps should do. What we do is have a single biomass equation that does not take into consideration subcellular compartmentation. In that, once the products have been made, they can locate to any part of the cell. Maybe we should be no, that's okay. Because the biomass equation is not looking at labeling patterns. The biomass equation is defining or setting limits on fluxes out of the metabolic network. And it doesn't matter where in the cells those reactions that are providing those precursors are located. The biomass equation is saying they are being withdrawn for biosynthesis, irrespective of their locations. So it's a good question, Kushboo. You've made me think again. But
thank you, Nick. And the next question is from Chandrakant Joshi. Imaging talk. If we have multiple organisms growing together, utilizing the same substrate, what are the possible ways, other than reporter proteins, to elucidate the central carbon metabolic fluxes?
It is difficult, which is why we have focused in our own work on reporter proteins, despite the immense challenges that exist in analyzing those. An alternative possibility that we should never overlook is a very simple-minded solution, which is simply physical separation of the components. So if there is any way in isolating individual units, so individual cell types or individual species in a bacterial co-culture, then that could be done. So for example, in maybe a plant rhizobial system, we can think of very exotic approaches based on reporter proteins, but an equally effective way may simply be to physically separate the bacteroids and the plant tissue and analyze them separately.
Again, quenching and other challenges would be there as well.
Yeah, well, quenching would be a problem if we were focusing on attempting to get the labeling patterns in metabolic intermediates. It's less likely to be a problem if we're sampling the labeling patterns in biomass end products.
Correct. Okay, one question on the subcellular fractionation. This is from Lisa. How do you define favorable conditions to not need subcellular fractionation? Potassium limitation seems not favorable. How can you exclude transport across organelles, especially into the vacuole? Example, sucrose or hexoses. Would it not be beneficial to combine those approaches?
Combine, I'm not sure which approaches we should combine. I'm sorry, Shyam, I've forgotten the beginning of the question.
So how can you exclude transport across organelles, especially into the vacuole? So I think the question correlates to including those those constraints, especially the transport across organelles, in order to define favorable or non-favorable conditions. If
Okay.
I'm getting it right, Lisa.
Yeah. Okay. So I would say with regard to defining or being able to define subcellular fluxes, we don't know whether the system is amenable to being able to get accurate determination of subcellular fluxes or not until we've done the experiments. So we appear to have been particularly fortunate with Arabidopsis cell suspension cultures in that there is differential labeling of hexose phosphate pools in the cytosol and the plastid, but that need not necessarily be so in all plant tissues. And indeed, Yair Shaka-Hill has evidence that in the developing embryo of oilseed rape, he has equivalence of labeling in sucrose and starch.
Yeah.
So you don't know until you've done the experiment. If the circumstances are unfavorable and you are unable to differentiate between cytosolic and a plastidic flux, then that will become apparent in the flux map because you will simply have very much higher confidence ranges for your flux estimates, and the flux through a cytosolic and plastidic reaction will be inversely correlated in individual flux determinations. So essentially, the flux collapses and is indeterminable.
Correct. Okay.
The secondary point, which I think is an entirely different one, which is with regard to, for example, metabolites moving across the tonoplast and accumulating in the vacuole. This can cause considerable problems because it can essentially mean either that when you extract the material and analyze the labeling pattern in a metabolite, it can mean either that you have to deal with a large pool of pre-existing metabolically inactive unlabeled material, but that can be included in the calculations using that cryptic T value. The alternative is that that large unlabeled pool could actually provide an additional input of material into the metabolic network and therefore influence the labeling patterns in your compounds. Again, you can include that in models just by introducing it as an additional unlabeled source. And that's frequently done in systems that are maybe slow growing, where there's a belief that there may be substantial protein turnover, in which case there's going to be a continuous input of unlabeled amino acids into the network from the breakdown of pre-existing proteins that were there prior to the introduction of the label. These are all factors that are amenable to inclusion in models and just reinforces my earlier point, which is that you need to think carefully about the details of the model you're using for flux analysis and not simply take a microbial model or one that you've knocked up in half a day.
Correct. Okay, great answer, Nick. Very detailed, and in fact, I think it captures most of the essence, especially with the plant systems, which are basically slow growing as well as with large pool sizes. Now, I thank you very much for such a excellent talk covering all the fundamental aspects of 13C MFA. You covered the tools, the key modeling considerations, as well as the subcellular flux analysis via targeted labeling. Thank you, Nick. And I hand it over back to you. You will be chairing the next session. While you introduce the next speaker, the audience can take a little bit of break and come back, maybe a couple of minutes. But you can introduce the next speaker. So Chandrakant, please share the next.
Okay. So Shyam, shall I begin introducing now,
Yes.
or shall we? In fairness to the participants, should we perhaps have a 2-minute break before I introduce Ashish?
Sure.
2
Let us
minutes.
wait for 2 minutes and then start. Ashish, sorry to keep you waiting.
Yeah, no problem at all. It was fascinating. I mean, it's thank you, Nick. So you already introduced, I think the first part of my talk is essentially your last question. The answer that you gave in the last question, and it's amazing. It's just awesome. So hopefully, I can sort of give more details, which can sort of you did such an excellent job, right? Just in that 2 sentences, you basically described what you have to do. So yeah, it's great. Yeah.
Oops. So why did it take me so long to say everything else then?
I don't know.
Okay, then maybe we should begin. So welcome everybody if you've returned. And it's my pleasure to introduce the first speaker of this session, Ashish Misra, who currently works at the Department of Biochemical Engineering and Biotechnology at IIT Delhi. His work focuses on a number of things, and currently his interests are on microbial metabolic engineering, and today he's going to be sharing with us his research on 13C MFA of poplar cell cultures under carbon and nitrogen availability. Ashish, over to you.
Ashish, are you online? Your video is off as well.
Sir, I think he had some issue with the internet connection.
Okay.
Okay.
We'll wait for a couple of minutes. Meanwhile, if Nick may permit, I can ask the previous one more question which was left while Ashish joins.
Okay.
That is, can FBA models be tested in 13C MFA or vice versa?
Yes, it's entirely possible to. An example of that was given by George yesterday.
Sorry, Nick. I think Ashish joined back.
Yeah,
Yeah.
I
Okay.
apologize. I was just answering your question, and I spoke for like 5 minutes, and then I realized the network had suddenly gone away. It was just so, and then I was frantically trying to reconnect, and it just wasn't reconnecting.
Yeah.
Sorry, I apologize.
No problem, Ashish. We introduced you already. Please continue. So you can
Okay,
share.
thank you very much. My apologies to the audience. It hopefully, right? I mean, fingers crossed. Hopefully, this does not happen during the talk. So, please let me know if my screen is being shared. Hopefully, it is. Yeah.
Yes.
Yes. Okay. Thank you very much.
That's good. Okay. Over to you, Ashish.
Thank you very much, Nick. So first and foremost, I would really like to thank George, Nick, and Shyam for organizing. This has been amazing. So far the whole of of yesterday, right? Listening to all of these talks, depicting these various aspects of how flux balance analysis can be applied to plant systems. Today we'll talk about C13 based MFA and how that can be used. So my talk is based from my postdoc work, which was done in the lab of Ganesh Sriram and Gary Pullman at the University of Maryland. At that time I was following Shyam's work a lot, and all of the papers that were coming out of Nick George's lab, with Arabidopsis and C13 labeling. And it was just very interesting, and we were essentially trying to apply it to sort of the next model system of trees, right, which was poplar. I'll present two parts. One will sort of, like I said, will expand on what Nick answered about modeling and reflux. And then the second part is really then looking at how different carbon-nitrogen availability affects flux distributions in plant systems, and specifically, we're talking about poplar. So poplar happens to be the model tree organism, and also it's an important biofuel crop because it's extremely fast-growing and perennial. So the thing that poplar and a lot of deciduous trees do in a very neat manner is that during leaf senescence, which is shown here, they are able to recycle. So I'm just trying to. I'll just get my pointer here. Okay. So during leaf senescence, when these leaves during the fall, when these leaves are lost, the plants essentially, these deciduous trees recycle a lot of their nitrogen back, and they are stored in the form of these proteins called bark storage proteins. So they get stored in the bark of these trees. And the work on this was done by Gary during his postdoc work. So here is an example. So this is a protein gel showing these specific bark storage proteins under short day conditions and long day conditions. So short day conditions is essentially when there are no leaves on the trees, and there is this storage protein, which is the bark storage protein that was that Gary found during his postdoc work. And when the trees are then exposed to long day, these proteins are degraded and essentially they disappear, right? Because they're essentially used for as storage for nitrogen, and this was the hypothesis of Gary, that they are used for storage of nitrogen during the short day conditions, which can then be used during the long during spring regrowth. Now this is even more dramatic, where this is the transition from the short day going to the long day. So this is week 1 of the long day, where the bark storage proteins are still there. This is the transition on week 2, where they're still there, and week 3 they are gone, right? So these proteins serve as important reserves for nitrogen, because carbon is obviously quite easily available to these trees in the form of they can easily use photosynthesis for fixing carbon. However, nitrogen ends up being their really key nutrient which they have to conserve. So this project was essentially to look at how carbon nitrogen availability changes metabolism of trees, and specifically poplar. And we applied a bunch of techniques. We used transcriptomics, proteomics, and metabolic flux analysis for trying to look at how metabolism changes. Now I'm going to talk about the metabolic flux angle. So Nick has already done a fabulous job of introducing carbon 13 labeling experiments, and I will just briefly go over this. So all of the labeling experiments here are going to be carbon 13. Right? So we are not looking at the transient phase. We're only looking at steady state labeling. So essentially what you do in a steady state labeling experiment is you feed a labeled substrate. It could be glucose or any other substrate that you're interested in. As the labeled substrate is fed to the cell, the cell metabolizes it, and it gets incorporated in the amino acids and all of the other metabolites in the cell. So traditionally, the amino acids, lipids, and other biomass components are the ones that have been analyzed for looking at the label on these. And from these, then you can do a bunch of mathematical calculations using metabolite balancing and using isotopomer balancing, coupled with optimization and statistical approaches to come up with predictions of fluxes and their error estimates based on the calculation, right? So it's essentially a fitting problem, a large-scale fitting problem. And eventually what this gives you, the experiment is it gives you a range of flux values along with certain error estimates. So that's C13 based metabolic flux analysis in a nutshell. Now the workflow is shown. This is just a quick slide from Gregory Stephanopoulos' textbook. This again just shows how you go about doing this, where after making the initial measurements of the metabolite pools that are available from experimental data, you sort of have a set of initial guess fluxes, and you take your network available from biochemistry. So you make your model. From that, you can formulate isotopomer balances. You take these flux estimates and you solve for the metabolite isotopomers. And then you go iteratively. It goes through this process and comes up with your best guess or your best fit estimates of fluxes. And that's the process of doing C13 MFA, right? So for doing the analysis on poplar, the first thing was, what we could was just setting up these isotope labeling experiments, and because poplar is essentially what we did was we did the analysis on suspension cultures of poplar, and because it's not possible to do this on the whole tree, or it wasn't at that time. I think at this point with advances in stationary labeling experiments, you can still you can do a little bit more than you could do about 10 years ago. So we did this on suspension cell cultures, and so I'll just talk about what we saw at the start. So the first thing was just trying out these suspension cultures. And this was a setup for the experiment, where initially we had some number of unlabeled cells. We fed it labeled substrate and allowed it to grow for a week initially. And then we just went about analyzing the labels on the amino acids, which were obtained from the proteins. So when we initially did this, we initially started off with what we had thought was 30% uniformly labeled glucose, but it ended up being 28% because of some experimental error. And here is what we saw, which was sort of strangely, which was strange. So what we had expected was we would essentially see the same labeling pattern across all of these amino acids, right? Because if you think about this, if there's glucose coming in and you are giving it enough time for the cells to grow, there is no reason for one amino acid to be differently labeled compared to the other amino acids. However, what we saw was quite different, right? Where some of these amino acids, so this is the expected labeling. So we found that some amino acids and some fragments of these amino acids were labeled to a larger extent than the other amino acids. Right? So alanine is labeled to a very high extent. Aspartate, isoleucine, leucine, lysine, and some of these other amino acids are labeled to a lower extent. And initially we thought maybe this is an experimental error, maybe something just went wrong. So we repeated it. This is across multiple replicates, we were able to see this. And we were stumped by this. I mean, you're not sure what was going on. So we then went ahead and did a labeling with what we thought was 100% uniformly labeled, but essentially it's 98% because of impurities. This is that's just how that's the manufacturer, right? They give you only a certain purity of this uniformly labeled carbon glucose, and we pretty much saw a very similar pattern where we saw a high labeling for some of the amino acids and very low labeling for a lot of the other amino acids, right? So some of these amino acids are, with label of 80%, and this is when you are feeding, fully labeled glucose. And again, this was puzzling to us because it would have made sense if we had seen similar labelings, right? Sort of like a straight line across all of these, which is what would have indicated that the initially present biomass was what was leading to a dilution of this label. However, what we were seeing was differential labeling across these amino acids. And so initially we thought, could it be because there is photosynthetic fixation of carbon dioxide, right, which is then leading to a dilution of the label. So what we did was we essentially took these plants, these poplar cells, suspension cell cultures, and we grew them in light and dark conditions. So these were green, even though they were suspension cells. They were green in color, and so we thought maybe it's photosynthetic fixation. And if you can see this, right, the differences between the photosynthetically or the cells that were grown in light, which are the ones in red, and the ones that were grown in dark, which are shown in green, was very insignificant. Right? Except for some difference in glutamate, there is really no difference across the amino acid labeling patterns. And we are again seeing the discrepancy where alanine is very highly labeled, and some of these other amino acids are labeled to a lower extent. So then the next question that we had was, so it makes sense that the initially labeled the initially unlabeled biomass, which is your starting biomass, obviously it makes sense that that's the one that's leading to this difference in labels across amino acids. However, we wanted to check, what we would see. So if you have this initially present biomass, right, which is not mixing with this new biomass that is getting formed because of the labeled substrate, what you would see is that all of this is unlabeled. So this would be represented by the M+0 peak in a GCMS. So essentially, it's completely unlabeled. And when you feed it completely labeled substrate, if everything else is getting synthesized only from the labeled substrate, then you should just see the peaks corresponding to these two, right? So this would be unlabeled, completely unlabeled, and this would be completely labeled, is what you would expect. However, the patterns that we were seeing were quite different, where we were seeing where we are seeing these intermediate fragments that were labeled, and it was different across the amino acids, right? So histidine, for example, has a lot more unlabeled amounts. Alanine is much lower, and it was different across these different amino acids. So if I was to just try to depict this pictorially, essentially this is what the problem is. So if the initial biomass and the newly synthesized biomass stay separate from each other, then you should just see those two patterns, just completely unlabeled and completely labeled. However, what we were seeing were these intermediate labelings of the amino acids. And so then we sort of scanned the literature, and at that point, we did not find anyone reporting on reflux, essentially of this mixing of the initial biomass with the newly synthesized biomass. Now, in microbial cells, on which a lot of the C13 experiments had been done, they grow really fast. So there is just a very low amount of the starting material, and the final label stuff pretty much overpowers the initial stuff. So it does not contribute to a significant difference. However, because these are slow growing cells, as Nick again has already mentioned, what we suspected was that something like this was going on, where the initial biomass and the newly synthesized biomass, they were sort of mixing, where the initial biomass was not sort of stationary, where it was just saying, I'll just stay here. But the amino acids in there were getting recycled, and not just the amino acids. Obviously, it would include the lipids, carbohydrates. But since we were only measuring the amino acids, that's the only information we had information on. Right? And so different amino acids were most likely getting degraded to different extents. And so we had to sort of change our model to look at what was going on in the system. Right. So here is what we did. We basically took the data from that 28% uniformly labeled, the 98% uniformly labeled, and 100% 1C13. So just glucose that was labeled at the first carbon atom. Right. And we performed these experiments parallelly. And then we fitted the data that was obtained to 4 different models. Right. So the first model was just based on taking the glucose and then just diluting it by the factor by which by the factor of the final to the initial biomass. Right. The second was done where the isotopic labeling was corrected based on the ratio of the initial to the newly synthesized biomass. And then the third was one where we just did it based on the amino acid composition, and we said, we'll just do it based on the initially present amino acid to the final present amino acid. So 2 and 3 are similar. 3 is just more detailed than 2. And then 4 is a really detailed model where what we said was, so if I was to show you the model itself, so generally in the model, the substrate is glucose, which is assumed to be coming in. This is the label substrate. And you then try to fit the labelings that are obtained on various amino acids. However, this 4th model is a detailed amino acid reflux model, which essentially allows a bunch of these amino acids via their degradation pathways to make some of these metabolites. So if the initial starting amino acid is unlabeled, you would essentially allow it to get degraded via its degradation pathway, leading to some unlabeled metabolite within the system. So that was our detailed model. So we had 4 of these models. And we fitted this to a typical compartmented, model of a compartmented cell, where central carbon metabolism, so glycolysis, the pentose phosphate pathway, and then the duplicated glycolysis in the plastid alongside the pentose phosphate pathway. And then a bunch of these metabolites leading to a biomass. Right? And here is what we saw. So this is just fitting of that data where the gray is basically the the second model. The green is the first model where the glucose is getting diluted. The blue data points represent one where we allowed the isotopomers to vary based on the different amounts of the amino acids. And then the orange is the amino acid back mixing model, where we allow the unlabeled amino acids to come back in and mix alongside the glucose, labeled glucose being uptaken. Right? And this is essentially a plot of the measured isotopomers against the simulated isotopomers based on the model and the best fit that they could make. So this is essentially just fitting. So Nick has already described this in great detail. And I'll sort of just show you that really the detailed amino acid model is the only one that that does a good job of minimizing the error between the simulated and the measured. All of the other models just do a very poor job, and it makes sense, right? Because if the process that's going on in the cell is that these unlabeled amino acids are getting degraded back and the labeled glucose is synthesizing stuff and they are mixing with each other, the other models obviously cannot capture that. And so we clearly see that that there's degradation of these unlabeled amino acids, which are then contributing and making biomass again. So this was quite interesting for us at that time because, like I mentioned, we hadn't seen this in the literature at that time, and I discussed this with Shyam initially, and he mentioned about having seen this, but I think at that time it was not reported. So in summary, we essentially saw differential labeling of these amino acids when we fed a labeled substrate, which was uniformly labeled, which was not expected. And essentially what we found was that you need a really detailed model to account for these labeling patterns. And this reflux of the unlabeled metabolite is very important in slow growing systems, which then needs to be accounted for in your model to be able to get good estimates on fluxes. Okay. And so this was in some senses a diversion, right? Because what we wanted to see was how different carbon nitrogen availability leads to differing fluxes within the poplar cells. So then we went back to that, where we wanted to characterize the metabolic landscape of poplar under different carbon nitrogen availability. And so this was the experiment that we designed after a lot of trial and error. So essentially just trying to see where we could see differences in growth rate for these different carbon and nitrogen availabilities. So we had two different carbon conditions. We had carbon that was growing at 20 grams per liter and another condition of carbon growing at 10 grams per liter. So this is high carbon, low carbon, and then high nitrogen and low nitrogen. And the way we, like I said, we went about selecting this was we wanted to see differences in growth rate across these conditions. We also wanted to make sure that by the end of our experiment, the carbon, which is in the form of glucose, was not close to exhaustion. The same thing for the nitrogen, because if it was close to exhaustion, essentially we would be seeing a change of metabolic state, and then you couldn't apply steady state MFA to characterize the system. So that was what went into the initial experimental design. And then we used combinations of this high carbon, low carbon, high nitrogen, low nitrogen. And we initially looked at the gross labeling, at the gross growth rates and the biomass compositions, and then we characterized the differences in the flux changes across these these cultures grown under different conditions. So the thing that the previous experiment had taught us was that we would have to essentially have detailed amino acid labeling patterns if we were to do a 1 week experiment, because there was very little initial starting biomass. And what we wanted to do was we wanted to sort of separate that out, because having a lot of these incoming variables just makes it may not give you very um, sort of perfect results in terms of being able to reduce the differences between these, because you have uh, too many of these other parameters which the system can change in terms of the unlabeled stuff that can come in. So what we decided to do was to do a longer labeling experiment so that we could essentially wash out in some senses. I mean, we are not truly washing it out. This is not a chemostat. However, we are diluting the initial biomass to such an extent that the final labeling pattern would be would have pretty much almost all of it being fully labeled. When I say fully labeled, the differences in the amino acids would not be significant. You wouldn't see these varying changes. Right, so we grew the unlabeled cells for over 7 weeks. So we grow them for 1 week. We added the labeled substrate. Then we again grow them for another week. We grew the labeled substrate, and we repeated this over 3 weeks. And the 3-week labeled cells were then characterized using the GCMS analysis to look at the isotopomers and then deduce the fluxes based on that. Okay, so let me go over the gross differences that we saw in the cells under these different carbon nitrogen availability conditions. So the first thing is, so let me just quickly sort of give a pointer. So the capital C's denote high carbon, capital N's are high nitrogen, low C's and low N's, small C's and small N's are the low carbon and low nitrogen. Right, so we saw that interestingly, under the high nitrogen conditions, but C not irrespective of the C, we saw higher growth rates for that. When there were low nitrogen that was available, the growth rates were similar irrespective of the carbon that was available in the media. Right, so essentially the cell growth decreased under low nitrogen availability. The next thing we wanted to check was what was happening to the biomass in terms of the carbon that was being left behind, right? So essentially what we were trying to check was what was the yield of this biomass on the carbon itself, right? And we found that, except for this low nitrogen condition where we did not, where our data is not, is a bit noisy, right? Because the R squared is not very good. We found that the yield of the biomass on the carbon does not change significantly across these carbon nitrogen conditions. However, what we find is the N itself, what it does is that the cells, when they have low amounts of nitrogen that is available, they consume low N to C. So they consume lower amounts of N, but they consume higher amounts of C compared to the high nitrogen conditions. So that is indicated by the residual nitrogen to the ratio of the residual nitrogen to the residual carbon in the media. So under the high nitrogen conditions, these have a higher slope, right, which is essentially indicating that the cells essentially took up more nitrogen. So really the experiment is proceeding in this direction, right? Where initially you have more carbon, you have more nitrogen and then it decreases. So the cells are consuming more nitrogen in the high nitrogen conditions in comparison to the carbon. And in the low nitrogen conditions, which are these two, they consume similar amounts of this nitrogen to C. So they are maintaining that ratio. So clearly this is indicating that the cells, as soon as they see nitrogen that's available in the media, they are essentially trying to grab as much of it as possible. Okay. The yield of biomass, though, on nitrogen, right, was higher under these under low N conditions. So again, these two are the high N conditions, and these two are the low N conditions. And then the biomass itself. So the low nitrogen conditions are basically trying to fix a lot more of the biomass by just taking up more of the C's. And so what you would then expect is that the low nitrogen conditions would contain compounds that have more carbon in them, right? Whereas the high nitrogen conditions, you would see the reverse. You would expect the reverse based on this. We actually analyzed. We are still looking at gross measurements. So we looked at the amino acid distributions across these conditions. What we saw was the amino acid, the overall protein content itself was higher for the high nitrogen condition compared to the low nitrogen condition, right? And then at this high nitrogen but low C, the protein is intermediate. It's not extremely high. However, the distributions of the amino acids themselves are quite different. And what we found was that under the low nitrogen conditions, we found that there was an abundance or comparative abundance of amino acids that had a higher carbon to nitrogen ratio. Whereas under high nitrogen conditions, we saw more of the amino acids that sort of had the reverse, which could keep more nitrogen and less carbon, right? So really, this makes sense because the cell is would be trying to take up as much of the nitrogen as possible. Right. And then we performed a flux labeling experiment. So this is the 3-week labeling experiment using a standard isotope labeling experiment. And we fitted this to the original model, so which does not have reflux. So this is a non-reflux model. So initially once the isotopomers were determined, the isotopomers corresponding to the amino acids, we wanted to see how significant their changes were across, so this represents the same amount of nitrogen but differing carbon, right? And here this represents the same carbon but the same carbon but different nitrogen. And so what we saw was that the isotopomers were different. The nitrogen a change in the nitrogen led to greater changes in between the isotopomers compared to when the nitrogen was the same, but the carbon was changed. The level of the carbon was changed. So this was just doing just a simple analysis of looking at the isotopomers and what differences were available. Now, after fitting the data, and I'm not getting into the details of that, so there's a lot of this computation analysis that goes into this before you can get a flux map. Then revealed a pretty interesting flux map. And so here the black is the high carbon, high nitrogen. The greens are the low carbon, high nitrogen. Blues are high carbon, low nitrogen. And the reds are the low carbon, low nitrogen. Right, so this is again the same model where we have glycolysis in the plastid. The TCA cycle. This is the cytosol. I apologize. And this is glycolysis and the pentose phosphate pathway in the plastid. I'll just point out some of these differences. So clearly, this major one that we saw was this great overexpression of the oxidative pentose phosphate pathway in the plastid under the high nitrogen conditions in comparison to the low nitrogen condition. We also found that the anaplerosis was affected, right, where the low nitrogen to carbon ratio led to an increase in the anaplerotic flux that was seen. And we also found was that in the model itself, we did need some acetyl-CoA reflux. So this was the only reflux that we added to the model. And we needed this, and we found that the low nitrogen to carbon increased the reflux of the acetyl-CoA that was there. Our paper actually discusses this in great detail, and again, I don't want to, for want of time, I don't want to go over every single detail. However, I'll just point out what we then saw with this oxidative pentose phosphate pathway upregulation. So clearly, the main function of this oxidative pentose phosphate pathway, as has been mentioned, is providing the reductant, specifically NADPH. And so then we looked at NADPH demand that was required by the cell for various aspects. Uh, so the solid bars indicate the NADPH supply that was estimated based on the fluxes that we had. And here we then show the NADPH requirement for various processes. So this is the NADPH requirement for the amino acid synthesis at the bottom. The NADPH demand for nitrate reduction is shown here, and then the NADPH demand for lipid synthesis by these cells. Right, and so there is a very good match. We found a very good match between the NADPH demand for these various processes and the NADPH supply that was obtained by the upregulation of the oxidative pentose phosphate pathway in the plastid. We also parallelly performed transcriptomic experiments on these cells that we had. To look at the gene expression data across these various conditions. And we saw significant changes in genes. So transcriptomic data for this has been done in Arabidopsis. I believe from, I don't remember if it was Shyam's paper or Thomas, I don't remember. However, yeah, so that was available. So we did this for poplar, and what we saw was again significant changes. So transcriptomic data always has a lot of changes in comparison to flux data. You just see all of these genes which are completely changed, and the fold changes are significant, significant. So these are just log 2 fold changes that are indicated here. So this is a log, this is basically a 32 fold change. This dark green is a 32 fold change that's being seen here. And so we found that there were differences clearly across primary metabolism, sugar synthesis, and amino acid. I'm just showing the ones that we have pointed out. We also found gene expression data across things that were sort of unrelated, what we had initially thought to be unrelated, where we found data of changes for cytochrome, genes for cytochrome as well, right? And we also found changes for the genes that are involved in, that are part of the photosystem. I'll get to that. Which was quite unexpected because these are cells which are not photosynthetically active. Still, we found changes in those. So just, we did find that the lignin and the aromatic amino acid genes are upregulated under the low nitrogen supply, which makes sense that these would be needed for making these aromatic amino acids. And then we compared the fluxes that we had, the changes in the fluxes to the changes in the mRNA level. So that's shown here. So there have been attempts in the past, and generally these don't match very well. They are just they match in some cases. In many cases, they just don't match. So what we found was it does match across some of these conditions. So here they are quite similar for the glucose 6-phosphate dehydrogenase. However, for some of these genes, they absolutely don't match, right? The patterns are quite different. So here there is almost no change in the flux. However, the gene expression patterns are quite different. So gene expression data really cannot be used as an analog for the fluxes. You really have to measure these fluxes and to be able to say if there are patterns at that flux level, changes at the flux level. So my point is about we found that there were significant changes in the photosynthetic and light capture genes, which was very unexpected. So really this is at the high nitrogen conditions, and we find that under the low nitrogen conditions, many of these genes are significantly downregulated in comparison. So we were again, stumped by this initially, which cell that's not photosynthetically active, why would it want to make these proteins which are involved in light capture. So our after a lot of discussion, our hypothesis was that it's quite probable that these are serving as nitrogen sinks, just like the bark storage protein. Under these artificial conditions of having too much nitrogen, possibly the cell is just synthesizing these and allowing these to store a lot of the nitrogen under these high nitrogen conditions. So based on this, we then came up with a hypothetical model, sort of under two conditions, which was the high nitrogen versus low nitrogen. So this is sort of a synthesis of of the quantitative flux data, and then sort of a qualitative model where we think that under the high nitrogen conditions, the cell essentially tries to take up this nitrogen, and it has to, for the nitrate especially, it has to produce a lot of the reductant in the form of NADPH so that this can be fixed. And then the amino acids that get made could get stored in the form of various proteins, and some of these could also be the proteins that are involved in the photosystem. Which is quite counterintuitive. However, these heterotrophic cells possibly do this, and this was what we thought was going on. Right? So in summary, carbon nitrogen availability and changes in those significantly and substantially during fluxes. And we saw varying correlations in gene expression data, which is expected and very well known. And we think that these light harvesting and the photosystem genes involved in photosynthesis most likely serve as nitrogen sinks under excess nitrogen availability, as we had seen. In terms of acknowledgement, this work was done in the lab of Ganesh Sriram and Gary Coleman. So Gary works in poplar cell. He's worked on poplar throughout his career, and Ganesh has sort of done flux analysis of soybean embryos, and so then this project was sort of an amalgamation of these two techniques which could be applied to poplar. Shilpa and Xiaofeng were the two graduate students. So it was Shilpa, Xiaofeng, and me. We worked on all of these experiments. And Maggie and Whitney were the two undergraduates, extremely motivated, extremely dedicated, who helped us during all these experiments. So it took a lot of sort of effort on everyone's part, and a sort of breaking your head against the wall, trying to figure out what's going on with the model to be able to come up with these. And I'll be happy to take any questions. Yeah, thank you.
Okay, thank you very much, Ashish. I can't see any questions in the chat bar yet, but I do. Oh, yes, there is one coming through now from Tom Williams. Thanks for a great talk, Ashish. Did you need to model catabolism of all amino acids? And how did you deal with those where there are multiple pathways, for example, lysine, or where the pathway isn't known, such as histidine?
Yeah, that's an excellent question, Tom. So we ended up choosing one of those. And I mean, obviously, we could have made the model even more complicated. So essentially, we looked at the amino acids that were the least labeled, and those were the ones that we sort of took them out of the system because they were just not getting recycled, and the ones that were getting really highly labeled were the ones that were sort of contributing back. Obviously, we could have made the model even more complicated, but you can just you can increase the number of the free variables, and you get an even better fit. But we sort of tried to do a compromise between that. And for the degradation pathways, we just chose the best ones that we just decided, hey, this is the one that's most likely going to work.
Okay, thank you. There's no more coming through at the moment, Ashish, but I have one quick question for you.
Sure.
Your culture medium contained both nitrate and ammonium, and so were these metabolized? were these metabolized equivalently? And did the relative rates of the metabolism of those two forms of nitrogen vary depending on the nitrogen source or availability?
Yes, that's again a very good question, and unfortunately, I do not remember that off the top of my head. But if memory serves me right, I think they were always finishing up the ammonium much faster than the nitrate. That's what I seem to remember. But if I also remember, I think we tried to just use one of them, and we just couldn't get the cells to grow. So we just had to go back to that. But again, just excellent. Yeah, very. Sorry, I don't have.
No, that's fine. Shyam, in fact, found a similar set of circumstances with the Arabidopsis cells where we were incapable of growing them on ammonia alone. Right? And there's one final question then from Manish. How computationally intensive does it get in modeling, especially in terms of the reflux reactions?
Yes, that's a good question, Manish. So I forget who said it, but if you give me four constants, sorry, not four constants. Give me four variables, and I can model an elephant. I forget who the famous physicist was who said this, but really, if you just increase the number of variables, you can essentially fit anything. So the thing that you want to do is to have as few variables and still give you a good fit. It gets a bit more complicated if you essentially allow all 20 amino acids to come in, and it does not make any sense if it does not add value to your model. So you have to do some sort of, use Occam's razor and then, say this is enough and just go with that.
Okay, so there was a subsidiary question then from Manish relating to that, which is, can regrowing on the 13C substrate in multiple cycles solve the reflux issue? So were you able to decrease the complexity of the model by cycling?
Exactly. So that's what we did. By cycling, we just had to have the acetyl-CoA come in because we still couldn't account for it even after 3 weeks of growth. Okay. And in the paper, we actually then compared the 1 week to the 3 week and showed how the 3 week could be, did a good job of sort of taking that 1 week and making it essentially reducing the complexity of the model, if you will.
Sure. Okay. Thank you very much indeed, Ashish.
Thank you, Nick, and thank you everyone for listening in. Thanks.
Okay.
Stop.
Right. Yeah.
Chandrakant, please.
That's great. Thank you, Chandrakant. So it gives me great pleasure to introduce the next speaker in this session, Ed Smith, who's taking time out from writing his thesis to attend this meeting. Ed has the misfortune of having both George and myself as his PhD supervisors, but despite that has made good progress. His thesis is about quantifying redox metabolism in plants, and today he's going to share his insights into non-stationary metabolic flux analysis. So over to you, Ed.
Right, now let's just share the right thing. Oh no. Right, can people see my slides?
Yes, yep.
Perfect.
Yep.
Okay. Great. Well, yeah, thanks for the introduction, Nick. Yeah, I'm doing my PhD with Nick and George, so part of the sort of group of people that have had to endure their supervision over the years. But yeah, it's a real pleasure to be here, and hear about all the really interesting work that everyone's been up to. We've had some really good talks so far. And hopefully I can keep you all interested telling you about isotopically non-stationary metabolic flux analysis in plants. So what I'm going to do is go over how we do this kind of experiment in plants, give you an overview of InstMFA, thinking about when you'd want to use this technique. I'll go through an example from my own work using InstMFA in plant cell cultures, and then discuss some of the latest applications of InstMFA to whole plants, before finally looking forward to the future of this method and how we can apply it more widely. Now, my aim for the talk is to show you what's involved in doing isotopically non-stationary MFA, and hopefully convince you that it's something that you could apply to your own work, and it can have some real benefits for what you might be trying to do. So we'll begin by looking at what non-stationary metabolic flux analysis is, how it differs to steady state analysis, and why you'd want to use it. And Nick's already primed us a bit for thinking about these things, but it can get a bit confusing when we're talking about these different steady states. So the first thing we need to think about is what is a steady state and what we mean by a metabolic or an isotopic steady state. So it's a metabolic steady state. It means that the fluxes are constant over time. And at an isotopic steady state, it means that isotopic labeling is constant over time, after we've introduced a label to the system. Now, if we're doing a typical steady state MFA, fluxes have got to be at a steady state, unchanging over time, and we wait until isotopic labeling is also at that steady state before we start to take any measurement. And at this point, the labeling patterns in the metabolites are going to be dependent on the fluxes, and we can therefore deduce what those fluxes are. But for isotopically non-stationary MFA, fluxes still need to be at that metabolic steady state, but we take our measurements of the labeling in the metabolites before the system has reached an isotopic steady state. And so then the dynamics of label incorporation into a metabolite are going to be dependent on the pool size for that metabolite. So if there's a big concentration, it's going to take longer for that metabolite to become labeled. It's also dependent on the fluxes through that pool. So if the flux is faster, then the metabolite pool is going to be labeled more quickly. And it also depends on the connectivity of the network. So where the precursors for that metabolite are coming from, and in turn, how labeled they are. So when would you want to use isotopically non-stationary MFA rather than doing an isotopic steady state experiment? So InstMFA has a number of advantages, and it can address some problems that steady state MFA just can't. And we have heard a bit about these from both George and Nick's introductory lectures. The main one, and that is super relevant when we're studying plants, is the ability to measure fluxes in photoautotrophic systems. Steady state MFA can't be used because if the only carbon source is CO2, then at an isotopic steady state, every metabolite is going to be fully labeled and not contain any useful information. Another benefit to InstMFA is the fact that you can do experiments with shorter labeling time courses to get the information that you need to actually calculate the fluxes. So if you're looking at short-lived or transient metabolic steady states, you can still analyze those. And finally, you can also achieve some increased sensitivity with InstMFA. You can investigate metabolite concentrations and also better resolve some fluxes like exchange fluxes or cyclic fluxes. But there are some additional experimental and computational requirements for doing InstMFA. So thinking about experimentally, you've got to take more samples at very short time points, sometimes down to seconds or minutes, and you've got to take multiple samples rather than just a single steady state sample. You've also got to make sure you're really effective at rapidly quenching metabolism, because small variations in that quenching time could affect the precision that you're able to measure fluxes at. And now, once you've actually managed to collect the labeling data for different metabolites, there's also additional computational demand, because we've now introduced a time component to all those calculations. So we're solving differential equations rather than linear equations, and therefore the computation takes much longer. And generally, you're going to have to be using parallel high-performance computing to do this sort of analysis. But don't be put off by that. Hopefully, I'll still be able to show you how we can do these experiments, and you know that there are some cases where there's really no alternative to using InstMFA. So we need to know how to do this technique, and I'm going to now walk you through an example from my PhD work where I was using InstMFA in heterotrophic cell cultures, and I'll try and highlight the different experimental considerations we'd have to make at each stage, and that you might be considering if you're thinking of doing these experiments. So as a bit of background, in my PhD, I've been investigating how we can quantify redox metabolism in plants. This is important for biotechnology, as reducing power is needed for both biosynthesis and maintenance processes, which are generally going to be the targets for any kind of crop improvement. Now, at the center of redox metabolism are the pyridine nucleotides, and in particular NADPH, which links the oxidation of substrates to where reductant is going to be needed for biosynthesis and maintenance. And so during my PhD, I've been interested in how these different potential sources of NADPH are able to meet those changing demands under different conditions. And we just saw an example of that from the previous talk, seeing that flux through the pentose phosphate pathway had to increase to meet the demands for fixing nitrogen in poplar cell cultures. So in this particular experiment that I'm going to talk through now, I was interested in how fluxes in central carbon metabolism respond to a sudden increased demand for NADPH. Could mimic something like an immune response or to a pathogen infection, or a sudden change in external conditions. So to increase that demand for NADPH, I used the drug menadione. It's a redox-active quinone, and it can both directly consume NADPH and generate superoxide free radicals, which then have to be removed by antioxidant systems, which also in turn consume NADPH. So adding menadione really whacks the system and increases that demand. But this increased demand for NADPH is going to be relatively transient. And so after adding that initial treatment, the system is going to adjust fluxes to meet that changing demand and only remain at a steady state, or probably a pseudo steady state, for a relatively short period of time. And so this is one of the cases where we really have got to rely on InstMFA, enabled to measure the fluxes during that short-lived metabolic steady state. So just emphasizing this point a bit more, if we were to try and do an isotopic steady state experiment in plant cell cultures that might look at labeling incorporation into proteogenic amino acids, we might have to do a labeling experiment for up to 5 days, maybe longer, to get to that isotopic steady state. That's going to be way too long for the kind of transient NADPH demands that I'm interested in. Using InstMFA, on the other hand, we can use labeling time courses as short as 20 minutes or so, and so we can capture fluxes during those more transient, short-lived steady states. And so we've got to use InstMFA for this experiment. So now let's think about how we're going to plan the experiment. What's the first thing we need to consider? And that's how we're going to get the data that we actually need to do the flux analysis. We've got to take samples over multiple time points, but which time points should we take, and what sort of intervals do we need to extract the metabolites? So in general, when we're measuring fluxes, the more data, the better, but you've got to think about the experimental effort of lots of time points. And so we want to think about how we can get the most information without excessive experimental effort. So the optimal time intervals are going to depend on the rate that metabolites incorporate label. But this can differ quite dramatically between different metabolites, depending on the pool sizes and the fluxes through those metabolites. And if you look at this sort of simple example model, you can see metabolite A is getting labeled very quickly from a 13C labeled substrate, but metabolite C is taking much longer to get labeled, in this case because it's got a much larger pool size. And so you've got to take a pragmatic approach to try and capture the labeling dynamics of different metabolites. In general, you're going to have most of the samples taken at early time points within a few minutes, but then also some later time points to try and capture those more slowly labeled metabolites. And in general, if you spread out the time points exponentially, you get quite a good compromise between those two things and being able to capture the labeling in different metabolites. In reality, what you'll probably have to do is an initial trial experiment to try and get the right sort of window and see how quickly things are labeled. And in my case, I did that and then settled on these 13 different time points, ranging from as short as 30 seconds going all the way up to 4 hours. So we know when we're going to take those time points, but we've also got to decide what we're going to feed our cells, which labeled substrate. Now, this decision is easy if we're looking at photoautotrophic metabolism. I'm looking at heterotrophic cell cultures here, so I've got some more options. I can feed them labeled glucose that could be labeled at different positions. And again, Nick mentioned this a bit in his talk. It's possible to optimize the precise best substrate or combination of substrates to use to get the most information, but this relies on already knowing the fluxes in the first place, which if you're just starting out doing an experiment, is not necessarily the case. And there's actually some sort of work still to be done here, actually exploring what is the optimal substrate to use for an INST-MFA experiment. We can't just take the lead from steady state analysis because it's not necessarily the same optimal substrates when we're doing a dynamic labeling experiment. But for my analysis, I went for using 50% 13C6 labeled glucose, which should in theory give a good distribution of label throughout central carbon metabolism. So we know what substrate we're feeding and when we're taking samples, so let's walk through the actual experiment that I did. I take heterotrophic cell cultures and grow them for 4 days until they're growing at a constant rate. Treat them with menadione to increase that demand for NADPH and give them 6 hours to reach a new metabolic steady state. And I spiked in the 30C6 glucose to 50% of the total glucose concentration, extracted metabolites at those 13 time points over the course of about 4 hours, and then analyzed them using LCMS. Now, the quenching and extraction method is really critical when we're doing INST-MFA because metabolites can turn over very fast in seconds, minutes. I went for using filtration and then quenching and extracting metabolites in one step with cold solvent, dichloromethane, ethanol, and hydrochloric acid. The quenching can also be achieved in a separate step to the extraction by adding cold solvent to cells. And generally, studies on bacteria or algae have used this sort of cold solvent quenching because it can be a bit easier to take lots of samples at short time points. But you have to be careful if you're using this method and make sure that metabolites aren't leaking from those cells during that initial quenching process. So once we have extracted those metabolites, we then need to quantify the isotopic labeling in them. And to do this, I used anion exchange chromatography and a Q-exactive mass spectrometer. And this method was really good because it provides excellent coverage of central carbon metabolites, and in particular, good separation of isomers that can often be difficult to separate, like the hexose phosphates here. Now, you can use sort of any method you like, other GCMS or LCMS methods for analyzing these central carbon metabolites. And it generally depends on what you have available. But the important thing here, and that can be slightly different to steady state analysis, is that we're going to need to measure more than just those proteogenic amino acids. We need to measure labeling in those central carbon intermediates, and so you need the system to be sensitive enough to measure labeling into those metabolites. So from that LCMS analysis, we can then calculate the isotopic label incorporation into each metabolite. Here I'm showing the average isotopic enrichment for each metabolite over time. Blue is unlabeled, red is more labeled. And this is a hierarchical clustering. So we're grouping similar labeling time courses together. And we can see that glycolytic intermediates down here at the bottom are labeled fastest, reaching a steady state after about 30 minutes. Things like organic acids and TCA cycle intermediates are labeled much more slowly, still not reaching a steady state even after 4 hours. And this sort of clustering is useful to check we've identified metabolites properly and that they associate together in the ways that we might expect. But you could actually also use this to identify unknown metabolites or unknown pathways by seeing which metabolites are closely associated with known metabolites. So now let's take a bit of a closer look at the actual labeling time courses for those individual metabolites, that data that we're going to feed into our metabolic model. And now here I'm showing the proportion of the metabolites that have incorporated different numbers of carbon-13 atoms. So for example, if we look at G6P, we can see this purple line is showing us the proportion that's got 6 carbon-13 atoms in it with a mass of N+6. And we can see that that's what's increasing the most relative to the other mass isotopomers, because we're feeding 13C6 labeled glucose. And in general, we can see that the labeling time points that we chose give us a good range over the dynamics of the labeling for each metabolite, and we've captured most of the important information in about 10 minutes. And we can see similar labeling patterns for intermediates of the pentose phosphate pathway and starch and sucrose metabolism. But if we look at TCA cycle intermediates, we can see that they're taking much longer to incorporate carbon-13. And as we've already heard in some of the other talks, this is because plant cells have got really large pools of these organic acids, often stored in the vacuole. And so it can take a long time for these pools to become labeled. And you can see these metabolites aren't reaching a steady state even after 4 hours, but we can still actually see the dynamics of labeling in those metabolites, which shows why it's important to have a range of time points. Now, before we get to doing the whole flux analysis, you can infer some information directly from these labeling time courses. And if we zoom in on malate, for instance, you can see that N+3 malate, so it's got 3 carbon-13 atoms in it, appears after just a few minutes. And the only way that this is going to be made is from the activity of PEP carboxylase generating N+3 oxaloacetate from the N+3 labeled PEP, and that's going to then form malate. And so you can already start to infer some information just from these labeling patterns. But now we want to take all that data and formally analyze it using InstMFA. The first thing we need to do is make a model of the carbon atom connectivity, exactly the same way as you would for an isotopic steady state analysis. You can work from previous models, reaction databases like Aracyc, and also textbooks. But you really need to make sure that these carbon atom transitions are accurate. Very small mistakes that might not seem that significant can have a big effect on the ultimate flux estimation. So to deal with a few extra features of plant cells, like subcellular compartmentation, we have to add a few more features to the model. So when we extract metabolites, we're mixing together pools from different compartments and only quantifying labeling in that mixed pool. It's going to be a weighted average of the labeling in the individual separate pools. What we can do is add pseudo reactions to our model to account for this mixing, adding the proportions of metabolites from different compartments as another free parameter that's going to be fitted by the model. And we can do a similar thing with metabolites where there might be a large pre-existing pool, such as those organic acids that are stored in the vacuole. And we heard about this from Nick, those G dilution parameters. So we've got our model of carbon atom transitions and our labeling time courses. The other input we need is some biomass outputs. And once we've got all that information, we can do the actual flux analysis. And in my case, I ended up with 4,418 separate mass isotopomer measurements. So that's across 17 different metabolites that have got multiple different possibilities for the number of carbon-13 they can incorporate, 13 different time points and 3 replicate experiments. And the important thing to have is more of those independent measurements than the free parameters in the model, so that we can have an overdetermined system and actually calculate the fluxes. So there's a few different software options for doing this analysis. To date, Inca is generally the main choice that's been used for InstMFA, and that's what I used here. And again, we've already heard this a few times, the actual process of estimating fluxes, but I'll briefly go over it. We input that metabolic model in the labeling time courses, as well as knowledge of exactly which isotopically labeled substrate we supplied to the cells. And we start the process by making that initial guess at what the fluxes might be, simulate what the labeling pattern would be, and then compare it to the actual data that we measured. Now, if it's identical on that first guess, then great, you've managed to estimate the fluxes straight away, but generally the software is going to find differences between the measured and the simulated data. And so it's going to update that guess and try again and again and again until eventually you end up with estimated fluxes that closely resemble the actual measured data. And so when I put all my data into this process, success, we're able to measure fluxes and central carbon metabolism in these heterotrophic cell cultures using InstMFA and just that single carbon-13 labeled substrate. But we were interested in what happens when we stress out our cells with menadione. Can we actually detect any changes in flux using this method in that transient metabolic steady state? So we can also generate a flux map for the stressed out menadione treated cells. To make things a bit easier comparing these two, we can overlay the two flux maps. And so here the thickness of the arrows is dependent on the flux in the menadione treated cells, and the color represents the fold change compared to the control cells. And we can see a number of differences, particularly around entry of pyruvate to the mitochondria, as well as some changes in the pentose phosphate pathway, increased flux through there and increased flux through plastidic malic enzyme. But before we get carried away thinking about these changes in flux, how do we know that these are significant and how confident are we in the actual flux measurements? So one way to address this is by doing Monte Carlo simulations to quantify the effect of measurement errors in the actual network structure on the precision of the flux estimates. And so to do this, we take our best fit flux map, simulate what the perfect measurement data set would look like, add noise to those measurements based on the experimental error to generate a new synthetic data set, and then use that to re-estimate the fluxes in exactly the same way as previously. And if we do this many, many times, we can end up with a distribution of multiple estimates for each parameter. And we can visualize all those different estimates using principal component analysis, which, as you can see here, shows a clear separation between the two groups. And we can also go a bit further and do a supervised method that try and maximizes the separation between the groups and identify the parameters which are contributing most to that separation. And so when we do this partial least squares discriminant analysis, we can see that the parameters that are having the biggest effect on separation between those groups are that pyruvate kinase flux and the anaporotic flux through things like PEP carboxylase and mitochondrial malic enzyme, whereas things like flux through the TCA cycle are having a negligible contribution to the separation between those two groups, and they're much more similar between the treatment and the control. So let's just take a closer look at those changes in flux around pyruvate. Here we can see that the actual flux distributions from the Monte Carlo simulations, the dark gray is the control, the light gray is the menadione treated cells. And we can actually see that the spread of the data and that there are clearly differences between the treatment and control. We've got a decrease in flux through pyruvate kinase, increase in flux through PEP carboxylase and mitochondrial malic enzyme. And so it looks like there's a restriction of pyruvate entry into the mitochondria, and the cells are compensating for that by importing malate or oxaloacetate instead to maintain the TCA cycle flux. And if we actually look at the individual labeling time course for pyruvate, we can see that in the menadione treated cells, so the dashed line here, the label is being incorporated more slowly than in the control cells, supporting the flux estimation that we get from the InstMFA. It's always encouraging to actually see how that raw data relates to the flux distribution that we get, as it can often be quite obscured if you've got a large and complex model. Now this particular inhibition of pyruvate kinase and diversion of flux into oxaloacetate and malate wasn't necessarily expected in these plant cells. There is some evidence that this occurs in mammalian cells. Oxidative stress can inhibit the pyruvate kinase as a mechanism to decrease flux through glycolysis and increase flux through the pentose phosphate pathway. And we do indeed see an increase in flux through the pentose phosphate pathway, as well as other changes that are associated with increased NADPH production. But in the interest of time, I'm not going to go into all of these in detail right now. But to sum up what we've learned from this example experiment, InstMFA was able to quantify fluxes in central carbon metabolism in these heterotrophic Arabidopsis cells. And we could actually quantify significant changes in flux induced by an increased demand for NADPH from that menadione treatment. And now, hopefully I've made this method sound feasible, but one thing to bear in mind is the computational requirements for InstMFA compared to steady-state metabolic flux analysis, and that you're really going to need access to high-performance computing to perform this analysis. A flux estimation in this particular model took about 4 hours, or a Monte Carlo simulation took about 2.5 hours. And if you want to do lots of restarts to find a global best fit flux map or thousands of Monte Carlo simulations, then the time it takes to do that can get really long. Fortunately, you can do these different tasks in parallel. And so if you've got lots of computing cores, it doesn't necessarily take that long, but you're going to need access to that sort of computing equipment in order to do these experiments. So that's one example of InstMFA in plant cell cultures, and that's comparable to what you might do in algae or bacteria or other single-celled organisms. But how can we measure fluxes in the leaves of actual plants? And the process is essentially the same as I've just described. We can feed plants 13 CO2, extract the metabolites, measure labeling over time, and then use that to calculate the fluxes. But to date, there's still very few examples of InstMFA being applied to the leaves of whole plants. And so hopefully it really won't take too long to give you a complete overview of the literature of InstMFA in leaves of plants. So a recent and really excellent example comes from Berkeley Walker and Yaya Shaka Hill's groups at Michigan State in America. And here the authors used single mature source leaves and an innovative gas delivery and quenching system based on a Lycor gas analyzer. And they were able to measure photosynthetic fluxes and identify that the pentose phosphate pathway is actually a source of CO2 release during photosynthesis. And I should also mention that Berkeley Walker is currently advertising for a postdoc in his group to do exactly this sort of work. So do get in touch with him if you're interested in these kind of experiments. And the other work that I should highlight is that this original example of InstMFA being applied to mature plants from Doug Allen and his colleagues. And here the authors used whole Arabidopsis rosettes, quenched metabolism with liquid nitrogen, did the InstMFA, and were able to measure photorespiratory fluxes that were quite difficult to measure using any other methods. So that's the two major examples of InstMFA being applied to leaves. And we'll just finish off the talk now by looking into the future of applying this method more widely to plants. And the main thing we need is really just more application of the existing methodologies to plants. But what would be really awesome is development of more automation or high throughput methods, which could actually be quite revolutionary for the metabolic engineering of plants. So at the moment, when we genetically engineer plants to try and improve yields, we make some changes and see if the plants grow bigger. But we really want to design, be more rational in those designs, and we're going to need to know exactly what's happening at the level of fluxes when we make those changes. And really, InstMFA is going to be the only way that we're able to do that in photosynthesizing crops. So in summary, hopefully I've convinced you that InstMFA is worth the extra effort, and sometimes it's our only option, either for short-lived metabolic steady states or photoautotrophic metabolism. InstMFA was effective at measuring fluxes in heterotrophic cell cultures, and for measuring fluxes in leaves of whole plants, InstMFA is going to be super important because it's the only way we can really get at that information. And finally, there's still lots to discover, and we really just need more people to start applying these methods to all the topics they might be interested in. So with that, I'd just like to thank everyone that's helped me out along the way, my supervisors at Oxford, as well as my colleagues, and in particular, Jingyu Fu from Berkeley Walker's group, who gave me some really useful advice and code on getting the high performance computing to work with the InstMFA analysis. So thanks very much for listening, and hopefully we've still got some time for a few questions.
Yes, thank you very much, Ed. Yeah, we certainly have time for a few questions. And there are some coming in now. There's one from Yaya Shaka Hill. who's asked, please can you comment on the use of simulated versus measured data as the starting values for Monte Carlo simulations? That's a nice and very precise nuance of analysis.
Yeah. So yeah, hopefully I'll explain this okay. Basically, for the Monte Carlo simulations, this is after we've already got that best fit flux map from the actual measured data. And what we then do is say, well, if this is the best fit fluxes, what is precisely the simulated measured data that would give me that flux distribution? And then taking that as our starting point, we add noise to that and then re-estimate the fluxes to get a distribution of fluxes or any of the other parameters based on that simulated data. So we're starting from the actual data saying, we're pretty sure these are the fluxes. How does any variability in that data affect that flux estimate? Hopefully that answers your question.
Okay, thanks. And a question from Kushboo Borah that's related to this in some ways, which is, I've lost it now, it's scrolled up. How do you define your MS measurement errors? Because this will have an impact on the Monte Carlos, but it's also important for the flux fitting or data fitting anyway. Were the errors experimentally derived?
Yeah, so I could talk about this for a long time, and I spent a while exploring this. So initially, when I first started out, I've got data from 3 replicate biological experiments, and I can look at the experimental error in the mass isotopic distributions between those replicates. But what I found when I used those errors in the actual flux analysis process is that for some time points, I suppose it's essentially down to chance, but there'll be very small standard deviations between those 3 biological replicates, and the model would end up biasing the fit to some specific time points. And so it turned out to be better to set uniform errors across the different time points. And I did that based on the actual measurement error, but also looking at scaling those errors until we achieve an acceptable fit. And I think I had it in the table there, that the errors that sort of represent the data, but still capture the variability and let us get an acceptable fit are between about 1.5, 1.8% of the actual labeling in each of those metabolites.
Okay, and in the interest of time, just a couple of maybe related questions from Berkeley Walker, sorry. And great talk, Ed. I think this is a comment rather than a question. Another thought for the future of flux measurements. We also think that a big area is to take these approaches into the field where plants actually grow and produce. I don't think there's any response to that other than assent and applaud.
Yeah, that would be awesome. Please do that.
But associated with that, and I don't know whether Berkeley wants to add anything to what I'm going to say here, but an additional comment is we have some initial data saying that the variation is not as bad as you would think. I'm not sure what that comment about variation relates to.
Might be in the errors, not sure.
I assume the errors.
But yeah, very happy to talk in detail about these things more. People just drop me an email and we can, have a detailed discussion about this, because yeah, there's definitely things to explore about what is the best way of defining the errors in those measurements and how does that relate to the fluxes you end up with.
Sure, absolutely. There are a couple more questions coming through on the chat bar, but I think maybe Ed will be able to address those just by responding directly in the chat bar. And in the interests of time, we need to move on to the final speaker of this session, which is Maneesh Lingwan. And Maneesh is a senior research fellow working with Shyam at IIT Mandi. His research focus at the moment is the reciprocal 13C feeding experiment strategies to capture metabolic crosstalk between host and pathogen. And today he's going to be sharing his thoughts on 13CO2 based mapping of plant stress. So Maneesh, the floor is yours once we get Ed offline.
Yeah, thank you. Thank you so much. So I am sharing my slides.
Okay.
Are they visible?
Yes, they're visible, and they're visible in the presentation mode. That's good.
Okay, thank you. Thank you so much. So hello everyone. So I'm
Manish, sorry to interrupt. You can turn on your video if possible.
yeah, I can
You forgot.
actually. How can I?
It's okay. Go ahead. Go ahead.
Okay. Can Chandrakant do that for me? No.
No, Manish, unfortunately, I can't turn on your camera. But if you see on the bottom, there is an option to start video. It says start video. Or where all the buttons are. So there is unmute, mute button, start video, share your screen. Yeah, now we can see you.
Okay. Okay. Thank you so much. So today I am going to talk about how 13CO2 metabolic mapping can be useful for the understanding plant metabolism. So if you look into the different approaches people are using to understand the plant metabolism, one is majorly the one is the metabolomics, and another one is 13CO2, where we both of those two approaches can give us the plant phenotype and help us to understand the plant metabolism. But when if you look into the plant, when it go into the biotic and abiotic stress conditions, the metabolism is modulated, and that can be a different class of metabolite, like a central carbon metabolites, sugar phosphate, aromatic, or some kind of a secondary metabolism, and in a very low level it can you can say that those can also modulate the phytohormones, and how we can capture them. So different sort of analytical techniques like NMR, HPLC, and GCMS, these analytical techniques can help us to define or to capture those metabolites in a cellular labels at the same, and capturing them is a one approach. You can call that metabolomics. But in a certain time, if you want to define a phenotype, so at that time 13C tracers strategies can quantitate the in vivo reaction rates, and then can better explain the cellular physiology. So if we're looking into the carbon and the plant is taking CO2, it may have a different fates, like in terms of making the energy, in terms of biosynthesis of other compounds, and in terms of nutrient uptakes, and in the developmental. At the same time, like carbon may transport it into a growing tissue and stores in form of a starch and transport in form of sucrose. So seeing that fate, that how someone explain the precise cellular physiology and the phenotype. So reminding and taking motivation and inspiration from the Calvin. If you look into he is the first one who have done take the 14 CO2 to understand the plant metabolism. But now we can see I have taken the motivation from and that the 13 CO2 can be used to understand the metabolism, and then dynamic labeling of 13 CO2 can precisely explain the metabolic pathways, and these literatures which are published nowadays motivate me to do the study more in those lines. So so my major focus of this what is happening and how we can do the system analysis of phototropic metabolism under ultraviolet rays, so that how we can do that for the first time. So for understanding the ultra UVB metabolism under how plant is coping with it, first we need to optimize different analytical platforms to capture the different class of metabolites, and at the same time optimizing the methodologies, their sensitivity play a major significant role. Then capturing those metabolites, the strategy, and doing some sort of statistical analysis is a one approach which we call the metabolomics. But at the same time, I have to go with the precise one, that we need to decide, and we need to design some experimental setup where we can feed the 13C to the plant, and then some sort of strategies to capture those metabolites. So this is the whole approach which I thought of to go and to understand the UVB metabolism in plants. So why should we care? Why should we care? Like what is happening with what is happening with the plant? How they are behaving? So if you look into there is a ozone depletion which can alter the ultraviolet levels in high altitude. In also where I'm living in Himalayas, that there's a high UVB rays. It can change the climate, and it can affect the forest and can affect the sustainable futures of a system. And if you're looking into the figures, it can affect a lot of and different sort of part of the earth. It can affect the plants and different organisms. But my focus is towards only the understand what is happening with the plants at present. So what we have sort of we reviewed and we studied that there are some morphological alteration in plant under UV, which few of you knows, and you know that it affects the germination, it affects the seedling growth. At the same time, it affects the rosette development, and also in a later development, it can affect the roots and the developmental process. So when we are going through those morphological alteration, we are looking into them in parallel. Our one group which in a collaboration, they have shown that there is some B box protein which are very important, and they play a very important role in light regulation, photomorphogenic regulation during the germination, and play a major role in a plant. And at the same time, they have seen that there is a one protein, B box protein, BBX31, which is playing a major role as a key signaling intermediate between the visible light and UVB light. And under UVB light, it is it is growing well, and it may be that time they propose that it is playing, BBX31 is maybe playing a major signaling role. So not confusing sort much of that but at the one side we are looking that UVB is altering the morphology and may be devastating for the plants. At the same time, someone is proposing that it is playing a major role and they are tolerant. So we have a question that how it is possible. So we have looked into the germination and growth of that. If you look into the figures, what is happening with the wild type and what is happening with BBX overexpressor plants. So when we are looking into the wild type plants, there is a they are less green. You can say they are less green. They may be a chlorosis, or maybe degradation of chlorophyll is there. But what is happening with B box overexpressor? So they are more green, and they are well tolerant, and you can say they have a different survival strategy to cope up with UVB. So what we have thought, that is there any metabolic survival strategy of B box protein, how they are, what is happening at the same time, what is happening with a wild type, why they are not surviving under high UVB lights. So we want to just uncover or decode that survival strategy of BBX31. So what we have done for the first, first we captured the photosynthetic compounds, how they are behaving, or what is happening with a B box under UVB conditions. So as morphologically we have proven, but as a biochemical we have checked that the level of chlorophyll is decreasing at the also the carotenoids levels, but it is lesser level of degradation in B box proteins, and at the same time the antioxidant levels like total phenolics content and antioxidant activity are the levels are higher in case of a B box protein. So and if you look into the phytol is well known for playing a major role in chlorophyll synthesis, but these levels are getting higher under UVB because it is not playing its part to make more chlorophyll, where it is not that case with the B box protein. At the same time, we have read that the level of dehydroascorbic acids are key signaling in an early case of UVB, which are enhancing under wild type, but that is not a case with overexpression of B box. So these all short of some biochemical analysis which we have done, and see that that there may be something which is happening with B box where it is playing major role, but not with the other plants. So we have done first some capture the metabolic labels through GCMS analysis we have done. And then the GCMS analysis tell us that there will be under white light conditions, maybe they are sharing some sort of metabolome with each other, that component analysis is showing us. But that is happening with the this is happening when we captured under the wide UVB light, they have a different set of metabolome. And looking into the heat maps and under different scores, we have look into that the phenylalanine, proline, asparagine, and succinic acid. Maybe those are the those metabolites are playing major role under growing of seedling stages. But this is just some small sort of information which are telling about metabolomics are giving us some signals to like maybe those metabolites are important. And then over discussion with Dr. Shyam, we thought of that like only just metabolomics is playing. Can we go with a dynamic metabolomics, that maybe we will get some more insight into or a precise biomarker under different light conditions, or like or a different exposure when we enhances the exposure of UVB, maybe we will get some more insight of UVB. So what we have checked, we have given UVB and white light of plants in 20-day-old plants into different exposure, like 2 hours, 4 hours, 6 hours, 8 hours, 10 hours, and all the whole period. We have a different set of time points, but the sort of information we are getting is more like more or less similar, that what we have seen is that there is a depletion in a TCA intermediates molecules. But if you look into the other some metabolites like valine, leucine, lysine, and phenylalanine, they are increasing after 1 hour of a UVB exposure. So in 1 hour of exposure, enhancement of those metabolites is something is questionable. Like is the plant is making more, like there are some de novo synthesis, or a plant is not utilizing those pools. Those are the questions which can be only be analyzed by the 13CO2 analysis, where we can say that yes, those pools are analyzing or not. And at the same time, if you look into the GO-GAT pathway, which is playing major role in a nitrogen metabolism, that sort of that is increasing after 8 hours of lateral stages. You can say that not initially, but later stages of higher UVB, they are a higher set of pools. But this is a dynamic metabolomics which are giving us some questions and some answers. But we have to take this. This give me the motivation to do. Like to make a plan for 13CO2 feeding. But 13CO2 feedings comes with its own challenges, and there may be some experimental challenges. Like if you look into the strategy of 13CO2 feeding, we have to feed 13CO2, and then there is a growth chambers which we need to feed 13CO2. We have to give the rapid quenching, and then do the some sort of analytical analysis, GCMS and LCMS, and those analytical comes with their own challenges, where we have to analyze different set of molecules like amino acid, sugars, organic acids, and we have to capture them and then do the processing, and comes to the metabolic modeling, which can precisely give us the flux map, and we can interpret something about under what is happening with the plant under UVB or not UVB, so or a control condition. So keeping that challenge in our mind, first challenge is that the design of 13CO2 feeding chamber, it is vital. Or so why it is vital? Because you don't need to, we need a control environment conditions to feed 13CO2, where we need to build, where we can feed, we need that that when we are feeding 13CO2 to the plants, there will be no dilution, no other factors are influencing the experimental conditions. So we need, and then in India, there's a very rare commercial designs are available where you can feed the 13CO2 to the plants and then do your experiments. And those I have gone through the all set of, they have a set of feeding chambers, and those feeding chambers have their own advantage. They have built their systems for doing their own experiments. So we have got the motivation from those, and taking all the challenges, we have designed our own 13CO2 feeding chambers. We established that facility, and comes with the comes that our systems can feed us to the any feed to the plants, and where I have taken the call zero and BBX studies. So looking into that system, they have its own advantage, where you can do the analysis, can feed the 13CO2 under white light, under UVB light, and you can control all the environmental factors, and you can capture the data like technical parameters. At the same time, this chamber will give you the levels of CO2, levels of temperature, humidity, and what intensity of light you are using, and what is the pressure of your chamber, that our systems can give you at the same time. So getting building that system and getting all the technicality, and proving that technicality, that they all are good, we have chosen to do the experiments. And for building this system and validating all the technical parameters, I just want to thank Dr. Shyam, Professor George, and Professor Nick, who helped me to calculate all the dilution factors, and the calculation over the how the dead volume matters a lot. And we have calculating all these things. And one more things I want to add up here, that simultaneously I just want to capture what is happening with the total starch and total sucrose under UVB light. So Coming to the, because if you look into the starch and sucrose, they are the two major photosynthetic products, which maybe in the, if we want to feed the 13CO2, we should know what are the steady state, or you can say that pseudo steady state metabolism at that particular time point. So under a white light is a different case, but when we are giving UVB light, then it may be going to be a different case, because plant is in a stress, and if your plant is in a stress, how it will it make a more starch, will it degrade, or maybe starch is not just accumulating and not utilizing further. And then if we have looked at, there's a degradation in a sucrose. There's a degradation in a sucrose, but there's an accumulation in starch. So that sort of, it is a more questionable for us, like how plant is making pseudo steady state under UVB or not. So keeping that challenge in our mind, we have feed the 13CO2 to the plants for the longer duration, from like in a 0 hour, 2 hour to the till the whole light period, and also and then left few plants for what is happening with the, like in a chase for the 24 hours, what is happening with the dark. We have left it, and then we have done the data analysis to check that is there any kinetic labeling or not. And then the data analysis is ongoing, and we have find that that there is a there's a kinetic labeling in a plant. So what first information it has given to us? The first 13C information that given to us, that there is a higher rate of protein synthesis in B-box protein under UVB light and under white light conditions. So because we have measured the proteinogenic amino acids, and have checked that there is a higher level of 13C incorporation in case of a proteinogenic amino acid, which reveals that there may be a higher rate of protein synthesis. Now we are capturing that with a fresh weight, and we also correlate them with a fresh weight, rosette area, and different photosynthetic biochemical labels, and they reveals that yeah, there may be a higher biomass and higher protein synthesis in case of a proteinogenic amino acids. At the same time, we have checked that what is happening with solubles under UVB and under white light. So if you're looking into the sucrose, then there will be a in white light conditions, in a proper white light condition, there will be a higher rate of incorporation, but it just diminishes, or there is a lower rate of incorporation, lower incorporation under UVB light. But the sort of information I want to tell, like what metabolomics can give you and what 13C mapping will give you, is that this one, if you look into what we capture through the metabolomics, is that there is a accumulation of valine under UVB light. Also similar information, that is metabolomics, that is dynamic metabolomics, which is telling us that there is a higher level of accumulation in all sort of genotype, like wild type, mutant, and overexpression line, which is tolerant. At the same time, that is happening also with the glutamate, which is enhancing under UVB light. But if you look into the incorporation of 13C under visible light, that although there is an increment in a visible light condition in the after different hour of 13C enrichment. But if you look into the UVB light, there is a very less, or you can say there's a no incorporation of 13C enrichment in valine. At the simultaneously, there is a slow labeling in case of a glutamate and lower enrichment of 13C. So and that information will you can say that in that case we have different set of interpretation, where we can say that maybe there is no de novo biosynthesis of those metabolites, or maybe a plant is using some intercellular pool of valine to cope up with the stress. So this sort of information is we can get from the metabolomics and 13CO2 mapping. 13CO2 mapping. So coming into the conclusion of the metabolomics, what we have done, so through the metabolomics, we have seen that there is a major metabolic reprogramming of plant under ultraviolet, right? And then at the same time, BBOX protein are tolerant to UVB light, and they have different set of, like, they have good photosynthetic levels, and they are the BBOX protein are making some like enhancement of some secondary metabolites, which may helping them to cope up with. But the main concluding remarks I want to give, and then there may be a future opportunities that we build up that 13CO2 parallel labeling system, which can help us to see what is happening with the photoautotropic metabolism, and then that setup can take up to the different plant microbe metabolic crosstalk. That will further talk in the next session. Will talk by the given by the Poonam and Dr. Shyam. They will, I think they will talk about how this system can help us to build plant microbial metabolic crosstalk. And this is my ongoing work, where I was trying to understand the photoautotropic metabolism under UVB, and I am open to take a few suggestions from the panel side and from the everyone, like how can I do the good for that, how can I understand more, and I'm open for any questions. And I just want to thank Dr. Shyam in that case, like he's my PhD supervisor, and consistently he's suggesting me. At the same time, Dr. Saurabh from Bhopal, who gave me to the BBOX protein lines, and consistently helping us to understand the developmental strategies of plant, and Professor George and Nick, who helped me during the like building up that system to feed up the like in stationary conditions of 13CO2, how we can see, and my lab mates, IIT Mandi, and at the same time, I just want to thank FluxMAPS 2021, who give me the opportunity to present my data sets, and to the everyone, and the one GCMS system, which I always like in the last four years, which helped me like the system day and night. I almost done more than 1,000 of samples there, and it helps me. So I just want to thank my system also. Thank you so much. I'm open to questions and suggestions.
Thank you very much, Maneesh. As he said, he's invited any questions, and we've got one immediately from Portia Singh, who asks, how do you manage the 13CO2 concentrations, the ppm levels, presumably in your feeding chambers?
Yeah, so we have, I told you earlier that we have a sort of some controllers where we can controlling the different set of oxygen levels, nitrogen levels, and CO2 levels. So those flow controllers, we flow with them in a some certain set of like volume per minute, and those volume per minute going further, mixing up in a one station, and then coming to further into a box where our sensors are making telling us that how much ppm at present is there in your system. So mixing up your different gas in a proper you can say proper concentration or proper volume, then you have to flow them in a chamber, and then sensor will tell you, like our sensor will tell you how much ppm is that. So that you can say that double we are pre-assuming that we are making a double step check for that much ppm we have. One is at initially where we are making them to mix up in a proper ratio with a proper flow, and checking with the sensors, where sensors are telling us that yes, that much of amount of CO2 in ppm you have.
Okay, there's a very helpful comment from Yair Shaka-Hill, and the relevance of this will depend on how exactly you're monitoring CO2 levels in your chambers. But Yair points out that depending on the sensor, erga measurements can be much less sensitive to 13 CO2 than to 12 CO2. So I guess, Maneesh, there are two questions here. Are you monitoring CO2 using an erga system? And secondly, do you take the relative sensitivities for 12C and 13C into consideration?
Yes, that I just want to make one point. We have run few experiment where there is some 12C dilutions earlier, and we have checked that there is a lesser level of 13C in our systems where we check the labeling in our metabolites. At the same time, when we are managed to dead volume in our boxes, that time we are getting the higher labels. And our sensors are just giving us that much of ppm. Like earlier we have done some experiment with 400 ppm. That is not exactly 400 ppm plus plus minus 50 ppm, or you can say that 450 ppm, and another one is around 800 ppm. And with when we are flowing it, but that in the first time of validation we are getting lesser level of 13C enrichment or incorporation in our metabolites. But when we are able to manage that volume and to flush out all the 12C out from the boxes and then done the experiment, then we have seen that there is a higher enrichment of 13CO2 in our metabolites.
Okay, thank you. Are there any other questions? There are none coming. Oh, here's another one. This is from Chandrakant Joshi, who asks, how different are the feeding experiments with whole plants compared to plant cells? And why did you choose whole plants?
This is some sort of autotropic or heterotropic condition you want to say? Sorry.
I think just did you consider doing these experiments? No, it's plants versus plant cells, he's saying. Did you consider using plant cells rather than whole plants for this work?
No. I just want to try with the whole plant because I just want to check what is happening with the UVB with the plants. Maybe I will take this into consideration. Yeah, but I don't know.
Okay. And then one final question then from Yogesh Pant, who asks, can your system be used to study the effects of high CO2 levels? And if so, how precise can it be?
Yeah, that answer I have given, I think already. We have checked it around at 400 ppm and at 800 ppm, and then we have seen that there are some labeling difference in between the higher CO2 and lower CO2 levels. Yeah, we can check it. Yeah.
Okay. Well, thank you very much, Maneesh. I think given the time, we should stop there. And so that's the end of this third session. The next session begins after a short break. It's scheduled to begin at at either 6:15 Indian time or 12:45 GMT, which is in approximately 10 minutes time. So I urge everybody to maybe get up, stretch their legs, have a short break, and resume in what is now 9 minutes time for the beginning of the final session. But I'd just like to take this opportunity to thank all three of the speakers in this first session of today. Thank you very much. And I'll see you all in 10 minutes time.
Yeah, thank you, Nick, for the break as well.
Hi, Kushboo.
Hi. Can you hear me well enough?
Yes, I'm just waiting for the signal to start from Shyam.
Okay. How are you?
Oh, fine.
Yes, let
I
us
hope
start.
you've been enjoying reading. Shyam, are we ready to go?
Yes, yes, George. Let us start.
Okay,
Yeah,
right.
Kushboo is also ready. Yes.
Okay, so a brief introduction then to our next speaker, Dr. Kushboo Borah from the University of Surrey. Kushboo did her DPhil in Oxford with me and Nick Kruger and Phil Poole. And she came to us from another IIT institute, not Mandi, but one of the others. And she now works in Surrey at the University of Surrey with Professor John Joe McFadden. And as you can see, she's working on Mycobacterium tuberculosis there. But for today, what she's going to talk about is work that she did in Oxford, I believe. And she's going to talk about the metabolic flux phenotype of the diazotroph Azorhizobium caulinodans. Over to you, Kushboo.
Thank you. Good afternoon, everyone, and good evening, I think, in India, yeah? So can I first please confirm if you can see my presentation?
Yes, I can see it.
Okay,
Yes.
thank you. So I would like to thank George for introducing me. And yes, I am going to talk about my work in PhD. And if there is time, I'll briefly touch upon how flux analysis can make major discoveries in infectious diseases, which I am using right now. I would also like to thank the organizers, Shyam and Nick, and the various organizations, UKIERI, Plant Sciences, IIT Mandi, who have brought us all together for this brilliant workshops. In my PhD in Oxford, I worked on nitrogen fixing symbiosis. So my work focused on identifying or measuring the metabolic fluxes of a diazotroph. A diazotroph is a rhizobia which can not only perform symbiotic nitrogen fixation with its host plant, but also as free living cultures. And this beautiful strain that I worked with was Azorhizobium caulinodans, and the type was ORS571. So when I will, throughout the talk, I'll be calling it ORS571, and hence why I have the number up here. The aim was to measure the metabolic fluxes that supports nitrogen fixation. And we did it in two systems. One was as free living cultures, and the other one as microsymbionts, which were isolated from the plant nodules, which you can see this globular structure here on the stem. This is the host plant, and also the structures nodules on the root. So this was the symbiosis I worked on, the bacteria on the left panel, and the host plant, which is a tropical legume plant. And it forms both stem and root nodules. And why is it important to study this? Because it provides most of the total nitrogen in the biosphere, and there is an increasing interest in improving this technology to non-legume plants for a sustainable agricultural systems, and also to improve food security. Now the equation here can say the nitrogen fixation involves reduction of atmospheric dinitrogen, and it's a very expensive process. And the tiny little rhizobians and the nodules does the job very well because it's got metabolism to support that. So, and as microsymbionts, there is another complexity because metabolism of both the host and the bacteroid will have to be synchronized so that it receives host nutrients and then adapts its metabolism and then fixes nitrogen and gives it to the plants. So I applied steady state metabolic flux analysis. I'll run you through the overall workflow that I adopted for 3 years during my PhD. So chose isotopically labeled substrate and developed a setup where the oxygen was controlled, the temperature was controlled for growing this bacteria, then measuring the isotopic distribution using GCMS, the standard workhorse, then constructed a metabolic network, as you have heard before yesterday and today, that the construction of this network requires knowledge of the literature, digging through the databases, and updated knowledge of what people have been publishing at the molecular level, which enzymes are active, and information on the genes and transcriptome. I did biomass measurements. So the biomass composition is important here because it informs us on how to calculate the biomass equation, which constrains the model. And then used INCA, which have been talked about. I think Nick explained it beautifully, and also Ed Smith about the isotopic network compartmental analysis, a MATLAB-based platform, which is very easy to use. And then obtained carbon flux distributions, and that describes the phenotype of the organism, either as free-living nitrogen fixes or as symbionts isolated from the host nodules. I'll just briefly run you through the aerobic setup, as in how did I define steady states, metabolic and isotopic steady states. So first of all, I chose, okay, which is the best carbon source to go for? Because within the plants, we know in the nodules of the plants, there are a number of carbon sources available. So I chose a bunch of them, the well-known, and then measured the doubling times. So the fastest was for succinate. So we said, right, okay, we go with succinate. And then also, I chose 2 different points in the exponential growth, early and late. And then at this 2 growth stage, measured a number of different biomass components: protein, lipid, RNA, DNA, PHB, polyhydroxybutyrate. That's a polymer in rhizobia, and it is a key polymer in nitrogen fixation. The exact role may be different in different rhizobial legume symbiosis. And here, I will show you that PHB synthesis does play an important role in nitrogen fixation in ORS 571. So the different biomass components at the 2 growth stages, they didn't show any significant difference. And going from that, we said, okay, cells are at exponential growth. So it is a metabolic steady state that is not changing significantly. And then also looked at the isotope incorporation at the late and exponential growth stages, and they all are strongly correlated. So we went ahead and said, okay, exponential growth phase culture between early and late can be considered steady state. Now, microaerobic, which is low oxygen growth. This was quite important for my work because the diazotroph will fix nitrogen only at low oxygen. So under free living conditions, as cultures, as bacterial cultures, I measured the doubling times of this bacteria at different oxygen concentrations. And it appeared so that 3% was the favorable one, throughout the range I tested, and then measured the nitrogen reduction in this bacteria at different oxygen concentrations again, and it appeared 3% oxygen is the most favorable out of the conditions tested. So we said, right, low oxygen, 3%, succinate, exponential phase. Now Bactroids, now these are a very different system to the free living cultures, the delegates, and also they have to be extracted from the root nodules, making sure that they are pure, just Bactroids, and not, and they don't have a big amount of the plant material surrounding it. So I used a protocol which was well established in Pool Lab. It involved a differential spin method, and got the Bactroids out, looked at both the stem nodule and the nodule Bactroids, and post-isolation of up to a 2-hour, they maintained their nitrogen fixation, that is reduction of atmospheric nitrogen into ammonia. And I'll just tell you how I measured this. So we used GC. So you put acetylene, and then you measure the amount of ethylene produced. So as it's doing the nitrogen fixation, that it's reducing the atmospheric nitrogen, it will also reduce the acetylene gas and make it ethylene. So hence why it's C2H4, that is ethylene production over time. And here at the bottom, this is looking at 13C incorporation in the Bactroids. That is just to say, if the Bactroids, after isolation from the nodules, can we detect any 13C signal in them? And the answer is yes. So we're very happy with that, and looked at this few amino acids and also PHB, our favorite polymer. And it did maintain, the isolated Bactroids did maintain 13C incorporation. After the experimental arrangements and after our satisfaction with those experimental setups, then we looked at the model construction. And one of the key points I would like to draw here is about model refinement, because in our model, we couldn't define the fluxes, especially of the anaphloretic node. So here on the left, you can see this is anaphloresis where substrates from the TCA cycle can be pulled out and dumped into glycolysis for use, or vice versa, can be fixed directly from the glycolytic substrate to TCA. So one of my colleagues and a former PhD student in Full Lab, he did a fantastic work in making many mutants of this anaphloretic node in the ORS571, and then tested its nitrogen fixing ability or acetylene reduction ability when the genes are knocked out. So DME here is the malic enzyme, which converts malic to pyruvate. And with this data, we were convinced that the DME is the essential out of the 3 or 4 anaphloretic node enzymes. Doing this was a tedious task, but also it helped us to gain confidence in the anaphloretic enzymes, which we should be using in our model. And this helped us to reduce the Gaussian knot problem, which is the identifiability problem of these fluxes which exist. And it's a well-known problem. So we moved ahead saying, right, out of all these reactions, DME is essential, refine the model, and then we did FluxMAPS. So we used INCA, like I said before, and this is the aerobic flux map of the bacteria as free living cultures. So under aerobic conditions, I gave it ammonium chloride in the media, and succinate as the labeled carbon source. And this is the flux map showing the utilization of succinate and the flux distributions when it's not fixing nitrogen and it's on the aerobic condition. So as expected, a bigger flux through the TCA and the anapleuritic node, because we're feeding dicarboxylate succinate. Going to microaerobic, in this condition, it was also ammonium chloride supplemented in the media at 3% oxygen, and succinate was the labeled carbon source. The aim of this exercise was to see what alone can low oxygen do. Forget about nitrogen fixation, but if I move the free living cultures to low oxygen conditions, what are the changes in fluxes? So the main thing to notice here is the flux to PHB, which is the polymer. So around 40% of the cell weight was PHB in this microaerobic cultures. So under low oxygen, whilst maintaining similar level of fluxes through the TCA cycle, the bacteria liked to channel carbons and dump it into the PHB. Then the third one was the microaerobic nitrogen fixing one, where there was no ammonium chloride in the media. Succinate was the carbon source, and it was at 3% oxygen, the low oxygen condition where it can fix nitrogen. And there were multiple changes, and the key one was the TCA cycle fluxes. There was a significant increase in the fluxes through the conventional TCA cycle. And then there is a greater flux or significantly higher flux through the DME as well, in addition to the PHB synthesis. So what it tells us that the flux through the TCA cycle is important for the free living ORS 571 when it's fixing nitrogen. So this was the beauty of 13C metabolic flux analysis, where you can clearly see the differences, and that too statistically significant differences under the three growth conditions. Here on the top left, this is the Bacteroid flux map. A Bacteroid is the bacteria which was isolated from stem and the root nodules of the plant. And what we found was a significant down reduction in the TCA cycle. So we were able to capture this difference in the Bacteroids. And in the figure here, it just shows an elaborated version of the different TCA cycle fluxes and how significantly higher they are in the free living nitrogen fixing culture as compared to the root and the stem nodule Bacteroids. So to take home, TCA cycle fluxes are important for nitrogen fixation. PHB synthesis, important or is affected, influenced by the microaerobic growth. Malic enzyme is important for nitrogen fixation. And I think a bit surprisingly, but reduced TCA cycle fluxes in the Bacteroids. When you think that in the nodules, it's much lesser oxygen than what I used for the experimental conditions, and yet the TCA cycle fluxes are significantly reduced. So this leads us to think that, that there is some kind of constraint that is imposed by the host in controlling the Bacteroid metabolism when it's inside in the nodules. And we have submitted a paper where we have tied up of flux balance analysis and 13C MFA to show or to conclude that provision of dicarboxylates from the host makes the Bacteroid adopt some changes in metabolism, and that TCA cycle is the one that was predicted from the FBA. And when we did MFA, we confirmed that prediction. So in this case, the FBA and the MFA results or the findings does match in my case. So I would like to thank George, Nick, and Phil Poole, who are my supervisors, and all the group members without which this work will not have been possible. I would like to also thank my scholarship, which helped me, which supported me throughout the study. So if I have some time, George, could I show some of the other work?
Yes, indeed, you still have time.
Thank you. So learning how to do MFA in Oxford helped me a great deal because I transferred that skill to working in tuberculosis and leprosy, which is a completely different field. And at the current state, I'm doing dual atom labeling, post-nutrient uptake, chemostat cultivation, and various forms of MFA, which I'll just briefly touch upon. So steady state MFA of TB pathogen was already established from George and Nick's and McFadden's collaborative work years ago. We just adopted the same way of doing things, but here we tested if you give different substrate to the TB bacteria, what does the fluxus look like? And this question was important because TB eats host cholesterol and acetate and glycerol. So we would like to find or target that reaction in TB bacteria, which then we can formulate a drug for to downregulate or to destroy that reaction in TB. So again, we have a chemostat setup where the growth of the TB bacteria is very well controlled. So this is a carbon limited chemostat. So we do the substrate measurements. We have the media feed going at a controlled rate, which then also controls the growth rate of the culture. We have pH probe, oxygen probe, and also air into the chemostat. So this is a very well controlled system because we were looking at metabolic fluxes at a particular growth rate. And this is important because TB is a slow growing bacteria. And in infections, when someone has a tuberculosis infection, bacteria in human replicates only in 7 days to 14 days. So it's very, very slow. Then we did steady state growth, like measuring the OD across 40, 50 days, across 2 months. Then we measured different 13C incorporation from the substrates and extended the metabolic network to include the methyl citrate cycle, because methyl citrate cycle is important for metabolizing lipids and cholesterol. So this work is just yesterday, it was accepted in molecular systems biology. We're very pleased with this. And just to show what I used INCA for this work and how powerful it is, on cholesterol and acetate, tuberculosis bacteria has got a significantly higher TCA cycle fluxus. And methyl citrate cycle it's forward or it's conventional on cholesterol acetate, but it's reversed in glycerol and tween. Tween 80 is a detergent. It's not a main carbon source. Here, glycerol is the main carbon source, but we use tween 80 to separate the lumps of TB bacteria because they like to stick together when we culture them. So we're currently developing 13C15N MFA, which is a dual atom tracking. Just to run you through, we're preparing this for publication. Again, chemostat setup. The difference is labeled carbon source, labeled nitrogen source. And the importance of this work is to find out the C and N fluxes, the C bond and the N bond fluxes, which are important in TB. So we use GCMS and also LC Orbitrap, because this is the only known system, as I know, can say which is 13C, which is 15N in the Isotopomer families. And then this work is done in collaboration with ULIC team. This is with Wykert, Katarina No, and using 13C Flux 2. And we have CM FluxMAPS showing amino acid and nucleotide metabolism. So just briefly, host pathogen metabolic crosstalk, which is a focus of this session. We previously used 13C MFA to ask which nutrients does TB take up from the human host cell. So metabolism is important for any organism, and for tuberculosis bacteria, which is MTB, is even more important because it has to survive and it has to cause disease. So it needs a lot of carbon sources, but what we didn't know, what are the nitrogen sources? So then we used human macrophage, where MTB infects. We infected them. Now did 15N isotopic labeling, which is amino acids with labeled nitrogen, and then did amino acid profiling and modeling. And this work was also done with the ULIC team, and we came up with 15N flux spectral ratio analysis, which I can speak to you in details if you want me to later after this talk or later in the session. And what we showed essentially was that TB bacteria here, if you look, all the red ones are the signals. There were signals for 15N in the TB bacteria when different substrates were provided to the host. So substrates first went to the host and then to the bacteria. So we found TB uses multiple amino acids and nitrogen sources, and that glutamine is the predominant nitrogen donor. And then touching upon the flux spectral ranges, we found that here glutamine and valine amino acids, their flux ratio was greater than 1, which from our modeling point, which meant that these are available in the human cell. MTB takes it up, and it can also be a nitrogen donor. If you look something like serine here, the minimum ratio is 0, means it's not available in the human cell for the TB. So this was a key work because we identified serine is not available to TB bacteria. We made a mutant of TB bacteria, which was unable to synthesize serine. It was called an auxotroph because it will grow only if you give serine in the growth medium. And then we chucked them into human macrophages, and we saw they were not able to survive in human cells, the mutants. The red line is the mutant here. So it says that serine biosynthesis is important for MTB. And using 13C MFA, we got our first target, and now we have a follow-up grant to make molecules against this gene, which synthesizes serine in TB. We have also done some work on leprosy, and here we just used 13C labeling. And here I just wanted to point out that 13C labeling experiments alone can also provide hints as to what the metabolism of the intracellular pathogen or a symbiont can be. So we deduced the isotopic profiles of the leprosy bacteria and compared it with TB. And the patterns, the blue and the green, they are different across some of the major amino acids. And using just 13C labeling data, we found that the anaphylactic node was differently used in the two pathogens. And so I would like to thank the mycobacterial team in Surrey, our current collaborators in ULIC, and people in Brazil who helped us with the leprosy work. I'm very happy to talk to you about in details, but I'm aware of the time constraints, so I would stop here. I'm very happy to take some good questions. Thank you.
Thank you, Kushboo, for that talk. Wide-ranging, taking in a number of bacteria. There's a question comes through. This one from Yair Shakahil. Thanks for a clear and interesting talk. The historic evidence for which metabolites are exchanged between the host and the bacteroid is indirect, mutants, and or disruptive perturbing. And I personally believe, therefore, insufficient, especially the recycling of amino acids. You have comments on this from your findings?
I'm just looking at the question again. Sorry, I didn't follow you.
Yes, well, it's a commentary and then a question.
Yeah. So why we did this? Because applying flux analysis and metabolic modeling to two organisms, like here, the human macrophage and the TB sitting inside the human macrophages, was complicated enough. And we pulled those two fractions out and and then adopted flux analysis by extending the model, saying, okay, these are the input fluxes from the macrophage. This is the labeling data from the nitrogen labeling experiments. I put it through the model. We'll try and simulate, and we find the best fit pattern for the uptake fluxes from the macrophage. And we did it a number of times, a number of runs. And it was inconclusive, as you saw from the list. For example, serine, which was 0, but then the maximum flux was above 1. So it was inconclusive. But it did have a hint that, okay, this could be the one that you can target. Whereas glutamate, valine, there could be potential exchanges between both the host and the bacteria during infection. So there could be a potential metabolite exchange between the host and the pathogen. That is why it is so difficult to make or isolate mutants of glutamine, glutamate, key amino acids in TB bacteria. And there exists no drugs that target this. So I agree that there will be exchange of nutrients between the host and the pathogen. And doing the exercise of flux spectral ratio analysis, we were able to identify 1 out of the 16 that was potential. So.
Okay, there is another question from George DiCenso, and he says there is evidence that malate may be the main carbon used by bacteroids in the nodule. Do you think your Azorhizobium results would differ if labeled malate was used instead of labeled succinate?
Oh, if I would think, it wouldn't differ, because it's a direct conversion from malate to succinate. So in Azorhizobia, we found that succinate and malate they had comparable doubling times, but succinate was lower. So if we go by our experiments and replicate some data, succinate is the one that we chose from our experiments. But I wouldn't think the flux pattern would be different if it was malate.
Okay, thank you. No more questions yet. We'll just see if anyone else comes through. No, it's all very quiet at the moment. Lets you off the hook a little bit. Yeah, here's another one. This is from Ed Smith. He says, thanks for the interesting talk and great to see you. For the combined 13C and 15N labeling experiments you mentioned, do you feed both substrates simultaneously?
Yes, I do.
Okay, that seems straightforward. And then a question from Bertrand Jacquier. Very interesting talks. I wonder if you could also follow your symbiotic fixation using 15N nitrogen, dinitrogen. This will also allow more clear indication of nitrogen fixation, because perhaps the acetylene test is quite imprecise.
What a great suggestion. I'm not in the nitrogen fixation field anymore, but it'll be a really important point to make, because you can have labeled nitrogen and then you can see how it's going and incorporated in the amino acids or any other metabolites, and then you can work out the rates. Acetylene test, yes, it could be a hit and miss sometimes, because it's not as we don't get exact numbers for the reduction assay, but it's it is one of the most easy things to do, well set up, and you get a quick readout of is it fixing nitrogen or not. So that's why we used it.
Okay, there's another comment coming from Tom Williams. Regards to experiments where you model glycine and serine metabolism, how does your model handle methionine, not methionine, MET-THF metabolism?
So our isotoper model is limited. We don't currently handle THF, FTHF metabolism. It's just serine to glycine, and then glycine, a backward reaction to serine, and then just a reaction going to MTHF.
Okay, I thank you very much.
Thank you.
And we'll move on now to the next talk in this session, which is going to be from Poonam. To what I know, she's going to be talking on metabolic flux analysis of the phytopathogen.
Thanks for the introduction. Okay, I'm glad and thankful to Dr. Shyam and Professor George and Nick for giving me this opportunity to present my work in front of all these experts and speakers. So by now we are quite clear about the paramount applications of 13C MFA with all the expert talks. So I'll add one more application to it, and the microorganism about which I'm going to talk about is Ralstonia solanacearum. So it's a devastating phytopathogen, and today I'll talk how using 13C MFA I decoded the metabolic phenotype of Ralstonia solanacearum, and how I found some bottlenecks in Ralstonia solanacearum to control this organism. But before talking about Ralstonia solanacearum, I want to emphasize on phytopathogens, because every year phytopathogens lead to around 25 to 30% crop loss, and among all the phytopathogens, bacteria account for around 27%. And if we talk about the top 10 phytopathogens, especially the bacteria, then these are the top 10 pathogens, and Ralstonia solanacearum ranks second among them. So Ralstonia solanacearum is globally distributed, and this organism is mostly affect the Asian and the African countries. The problem with this organism, which is a major concern, is that there is no effective control against the organism, and the farmer has to eradicate the entire field to stop the spreading of the organism. Another thing is this microorganism infect more than 450 plant species and mostly the crop plants. The bacterial strain F1C1 that I'm working on is isolated in Tejpur University. So here I want to acknowledge the bacterial strain. And so what this bacteria do is, using chemotaxis or through bones or natural openings, this microorganism enter inside the pathogen. And once it enters the root or any opening, then it moves towards the xylem of the plant. And inside the xylem, So this xylem, this is a very crucial step for the virulence. So inside the xylem, bacteria stop its mobility and it start colonizing. As the number of bacterial cells increases, it clogs the entire xylem. Also, it produces exopolysaccharides for biofilm production, and with both exopolysaccharide production and the bacterial cell, it clogs the entire xylem of the plant. And this clogged xylem acts as a barrier for the water conduction, which leads to wilting of the plant and ultimately the death of the plant. So there are a number of virulence present and produced by the bacteria. But there are no such metabolic and understanding the metabolic phenotypes. So here is the answer. So it's a very good work, excellent work done by the Dr. Catalin Allen from University of Wisconsin. So there they have represented that how there is a trade-off between inside the host that the organism has to distribute its energy in such a way so that it can produce virulence factors. Simultaneously, it can overcome the host adverse conditions and can grow properly inside the host. So two factors play a major role. One is metabolic versatility and another is substrate specialization. So there are microorganisms which are dependent on special substrates for the virulence factors. For example, in this figure, the yellow one, so this substrate is special for the virulence production, whereas the another one can be used for the growth as well as for the virulence. So the bacteria has to use the metabolic pathways in such a way so that it can grow properly, proliferate, and also produce virulence factors. So that is the main reason why I want to understand the metabolic phenotype, because if we know the metabolic networks and the metabolic pathways, then we can think of controlling the organism by targeting those particular pathways. In case of Ralstonia solanacearum, as Ralstonia reside inside the xylem of the plant, and xylem is a nutrient poor region. And another thing is that there is a continuous flow of the water and the nutrients. So we were interested to understand the metabolic phenotypes, and for that, first we thought to understand the metabolic phenotypes in very similar simple conditions, which were minimal media condition where glucose was the only carbon source. And the other condition was mimicking media condition, where we thought to mimic the conditions which are present inside the tomato xylem, and then to find out its metabolic phenotypes, that what are the features or metabolic networks are majorly active in mimicking conditions. Another step where we want to check the metabolic phenotypes is in planta condition. So in today's talk, I'll talk about minimal media and mimicking media conditions, and the in planta conditions, what is our progress and what are the challenges we are facing. So Dr. Shyam in next talk will explain about that. So in order to understand the metabolic phenotype, I have used 13C MFA approach, and I need not to explain much about this because in the morning, Dr. Nick has explained very well about all the 13C prerequisites. So I'll only talk about the steps that I have taken care. So in case of minimal media condition, I have used parallel labeling approach. There I used 113C, 1213C, and a combination of 1 plus U and U13. Whereas in case of mimicking media condition, with all the amino acids and the components of xylem, along with that, I fed the system with 13C glucose and 13C glutamine also. The glutamine was positionally labeled first and the fifth carbon. So I grew these cells in these conditions, minimal and mimicking condition, and then I grew them till they reaches to the pseudo steady state condition, which is the mid exponential phase condition, and then harvested the cells. Then I did cell hydrolysis, and then after TBDMS derivatization, I got mass isotopomers. Then I use the amino acid mass isotoper values to map the central metabolic pathways. So in case of Ralstonia solanacearum in minimal media condition, using these amino acids, I mapped the central metabolic pathways using retrobiosynthetic approach. And this work we have published last year in M Systems. So if I talk about ED pathways, so in this study, we found that in Ralstonia solanacearum, ED pathway is active, whereas glycolysis, which is normally active in almost all of the organisms, is absent in Ralstonia solanacearum. So if we focus mainly on the serine label, so X here, X is 12C carbon. One is where one, like the first glucose, first carbon of the glucose is labeled. 1, 2 is when first two positions of the glucose are labeled. And U is when universal label, it's like 12, 13, sorry, 12C only. So when 13C carbon enter in the system, so then the first position of glucose will enter in the first position of pyruvate according to our analysis. But if glycolysis is active, then according to glycolysis, the first carbon would result in label on glyceraldehyde 3-phosphate, G3P. But that was not the case in case of Ralstonia solanacearum. Similarly, with 1,2,13C, ideally in glycolysis, we should have label in glyceraldehyde 3-phosphate and subsequently in serine. But that was not the case. So the only possibility of unlabeled glyceraldehyde 3-phosphate is via ED pathway. Because in ED pathway, the first position, first 3 carbons ends up in pyruvate, whereas the last 3 carbons ends up in glyceraldehyde 3-phosphate. Similarly, we can figure out using alanine labeling also. So in case of alanine, if the first position of glucose is labeled, then by glycolysis, the third position of pyruvate should be labeled. Whereas by ED pathway, it's the first position of pyruvate or the alanine that should be labeled. So this 13C, the position I have explained here with using MID values. So let's suppose this is the first position of glucose, which is labeled, and glucose is entering by glycolysis in the system. So there should be label in glyceraldehyde 3-phosphate and the third position of pyruvate. So third position of alanine should be labeled. So if we focus on this graph of alanine, then alanine 260 fragment where all the 3 carbons are present, we have M+1 label. And similar with the ED pathway also. Now the question is about its position. So for this, we'll target alanine 232 fragment, which has only second and third carbon, and there's loss of first carbon. So first carbon will lost, then the alanine 232 will be unlabeled. And if the third carbon will lost, then there should be label, but which was not the case. So this clearly indicates that only ED pathway is active, not the glycolysis. Then we found that in case of oxidative phosphate pathway, so here also, when first carbon will enter into glucose 6-phosphate and then glucono 6-phosphate and ends up in 6-phosphoglucanoate. From 6-phosphoglucanoate to ribose, there must be a loss of first carbon if this pathway is active. So ideally, we should get unlabeled histidine, but that was not the case. We got M+1. So from this, we could say that this pathway is not active, whereas the label is coming through via this non-oxidative pentose phosphate pathway. We further confirm this using 1,2,13C. Here also, again, if there is loss of first carbon, the ribose should be M+1 rather than M+2, but we got M+2. So which is another indication that oxidative phosphate pathway is absent, whereas the non-oxidative pentose phosphate pathway is present in Ralstonia solanacearum. So this was about in minimal media condition. Then we formulated xylem mimicking media. In xylem mimicking media, so we constructed media based on literature, the xylem sap composition. So we fed the system with different amino acids and also with labeled glucose and glutamine. And using retrobiosynthetic approach, we mapped its central metabolic pathways. Here you can see the label is very less comparatively to the minimal media conditions. So that means that bacteria is preferring to use the amino acids which are provided in the media rather than using glucose to de novo synthesize amino acids. But still we got label to confirm the ED pathway. So from here I'll explain 1 to 13C labeling in ED pathway. So if 1 to 13C glucose is fed to the system, then we should get first 2 position of alanine labeled. So ideally in alanine 232 fragment, there should be M+1. But if glycolysis would be active, then we should have got M+2. So in case of xylem mimicking conditions also, we confirm that it is the ED pathway, not the glycolysis. Then another serendipity happened, and while I was running GCMS, I got a peak of 3-hydroxybutyric acid, which is a precursor for polyhydroxybutyric acid, a bioplastic. And we were keen to know that how glucose is coming to 3-hydroxybutyrate and what is the NADPH source here. So from this study, we found that ED pathway is act as a NADPH source for 3-hydroxybutyric acid rather than glycolysis. How? Because there is loss of first carbon here from pyruvate to acetyl-CoA in the form of CO2. So ideally, if ED pathway is active, we should get unlabeled 3-hydroxybutyric acid and M+2 label with 1 to 13C, which was the case. So from all these studies, we could say that ED pathway is majorly active and non-oxidative pentose phosphate pathway. But first we thought that these results are for our own bacterial strain and not for the other strains. So just to confirm that this is the case for our strain, so we did comparative genome analysis and thanks to our in-house tool by Chandrakant G2KO. So there we used the G2KO to compare bacterial strains, and I compare 86 Ralstonia solanacearum strains and 10 Ralstonia species to compare the metabolic pathways. And from the comparative analysis, I found that key pathway genes like phosphofructokinase, which is a regulatory gene of glycolysis, and glucanoate dehydrogenase gene, which is another regulatory gene for oxidative pentose phosphate pathway. So both these genes are absent, and all the genes required for the ED pathway are present in among all the Ralstonia solanacearum species. From the pathway mapping, we confirmed the pathway activities. Now the next step was to quantify the fluxes, that how much flux is going via which pathway. So for that, first thing I did is biomass estimation. I estimated DNA, RNA, and all the possible biomass constraints I could estimate. And then I formulated the network model from the date and included 133 reactions. And then I use Inca software for flux estimation. And this is the flux map that I got in minimal media condition. And you can see that ED pathway is majorly active, and there are anaplerotic reactions are also active, whereas the pentose phosphate pathway, upper glycolysis, and glyoxylate shunt are completely absent. Now the next question was whether with these network model and metabolic pathways, whether this bacteria is able to fulfill its metabolic energy demands or not. So I calculated the ATP, NADH, and NADPH values, and interestingly, this bacteria produce more NADH and NADPH, possibly via ED pathway, to suffice its demands. And so from this, we could say that this ED pathway can act as a bottleneck to control the pathogen. And keeping this hypothesis in mind, we used some commercially available ED inhibitors, which are pyruvate analogs, and it's bromopyruvic acid and 1,4,2,4-dinitrovenzene. And I used them against the bacterial culture. And I found significant zone of inhibition, which proves that if we use ED inhibitors, we could control the organism. Then in our lab, one of my colleague is working with essential oil, Mr. Maneesh, and he has done metabolic profiling of all these Himalayan essential oils. So we thought to try these essential oils against the pathogen and to find out whether these essential oils can act as biocontrol for the pathogen. So you can see in the figure, when we are using essential oil and infect the plant, then there is a growth of the plant compared to the other plants, which where we are not using essential oils. So this could be one direction where we can think of in future of using essential oil as biocontrol. And also we are thinking to use some metabolites, or to find out some metabolites which specifically inhibit the ED pathway and to control the pathogen. Then another piece of work from Katie Allen group, there they have presented that because xylem is a very nutrient poor reason for the microorganism. So how this organism survive inside the xylem of the plant? So we found they have stated that Ralstonia solanacearum manipulate the host plant to produce more nutrients, and which that organism can use for its survival. So they have stated few metabolites and organic acids like putrescine, then alanine, and some organic acids. So this is a good direction for us to understand the metabolic phenotypes in planta conditions. That how are what are the metabolic pathways are active in planta condition. So just to conclude my today's presentation, I would like to state that in minimal media condition, we have successfully mapped the central metabolic pathways. We got metabolic flux also 13C MFA in minimal media condition, and from both the studies we could say that ED pathway is majorly active, and that can be used as a bottleneck to control the pathogen. Whereas in case of mimicking media condition, from 13C mapping studies, we could say that ED pathway is active. Now we are in process to get 13C FluxMAP so that we can we can be 100% sure that under mimicking condition also ED pathway is active, and then ED pathway can act as a bottleneck to control the pathogen. But this presentation will be incomplete if I'll not acknowledge the people behind this story. And I really want to thank Dr. Shyam for guiding me through and supporting me throughout the journey. I also want to thank Professor George and Dr. Nick for refining my FluxMAP, and my collaborators from Tejpur University, who have kindly provided me with bacterial strain. Then Spark Project, FluxMAPS Biolab, IIT Mandi, and of course my friends. Thank you.
Thank you very much, Poonam. Very clear talk. Let's see whether there are any questions going to come in on the chat.
Yeah.
We're just waiting at the moment, but while we're waiting, how close are you to getting a flux map for the xylem limiting conditions? So it's almost ready.
Sorry, Professor, could you please repeat? What's the question?
I was just, while waiting for questions to come through, I was just wondering how close you were to completing the flux map for the xylem mimicking conditions. I don't think you're hearing me.
Sorry, some network issue. Okay. Yeah, so I'm really very sorry, but could you please?
No, let's go and look at the questions in the chat because something has come through,
Okay.
which you can probably see. It's from Ed Smith. It says, thanks for a very clear talk. Do you have any ideas which processes might be consuming the excess NADH and NADPH that wasn't required for biosynthesis? Could this be required for some virulence mechanism?
For now, I'm not very sure that in what processes it is consuming excess of NADH, but yeah, for NADPH, NADPH is required for PHP production, and this bacteria produce PHP indeed. So this could be one reason why it is producing NADPH, because PHP act as a carbon source under adverse condition for the plant. Sorry, for the bacteria inside the plant.
Okay, and here's a question. What advantage does the ED pathway provide to the organism over glycolysis?
Although in glycolysis it produces 2 ATP and ED pathway produce 1 ATP per glucose, but if we see the number of metabolic reactions or metabolites in between, I would say that in ED pathway there are less reactions or less metabolic intermediates, so bacteria need to spend less energy on producing them compared to the glycolysis where number of steps are more. So these are again some hypothetical studies which are there in the literature, but no experimental validation of that.
Thank you. Then there's a question come through from Kushboo, who says, what proportion of PHB and lipids did you measure in the bacteria?
So in PHB, in case of Ralstonia, PHB was around 2%, and the lipid content was approximately 3 to 4%.
Seems very clear. Question from Bertrand Gaquier. Very good talk. Did you identify molecules from essential oils that are protective for tomato plants? And will these molecules not be toxic to humans?
So for now, we haven't identified any molecules. We just did one small study that whether essential oil can help the plant to grow against the bacteria or not. But yeah, if we are, I mean, we really need to check it whether these are not producing any toxic compounds, or we haven't done any such study for now.
Okay. Ashish comes through with a comment, great talk, and then says, does the organism grow aerobically or anaerobically in its natural conditions? And also, does the excess NADH get converted to ATP?
So this organism can grow aerobically properly, and inside the plant the conditions are not very aerobic. Then these are slightly anaerobic condition, but it can grow properly under those conditions as well. And if the question is, does the excess NADH get converted to ATP? So ideally NADH can convert it into ATP, but whether this bacteria is producing or not, I mean, I cannot say anything about this.
So
but yeah, NADH can be converted to ATP.
In principle, yes. Okay. Any more questions coming in? It doesn't look like it. In which case, thank you very much, Poonam. Very good talk, very clear.
Thank you, Professor George.
We'll now see what your supervisor's got to say, because I believe the next talk is from Shyam.
Yeah.
Right, so on the screen now, you can see the biography of Shyam, who is, of course, well known to us all now, having run this symposium for a couple of days. He did do his PhD in plant sciences at Oxford with me and Nick Kruger. And he then did postdoctoral work in Oxford with Lee Sweetlove, and then moved on to the University of Bath, where he did some microbial work. And then he's ended up, as it were, in IIT Mandi. And he's built a group around his interests in flux analysis. He's going to talk to us today on a topic which I'm just pulling up to check. I've got it down as plant microbial metabolic crosstalk, challenges and recent advances. So over to you, Shyam. I'm all agog.
Thank you, George, for the introduction. I will share the content in a minute. May you please confirm if you can see my slides.
It's there. You're ready to go.
Okay, thank you, George, and everyone who, all the speakers as well as the attendees. I'm going to speak about, share our current work on plant microbial metabolic crosstalk. Mainly, I'll be focusing on the challenges and some of the advances which we achieved in the recent past. Before entering into the topic, I would like to briefly give a background about the group research activities which we are undertaking. I joined IIT Mandi in 2015. Since then, I've been, along with my scholars, we have been investigating on two parallel streams. One is our interest in relation to photoautotrophic metabolism, mainly in plants. We took up the approach of easy metabolomics initially, but gradually we built on that, and currently we ended up building a prototype for undertaking studies using 13CO2. Probably that will give us give us directions towards instationary flux analysis, which would be more relevant for agricultural applications, especially under the photosynthetic conditions. And the second parallel approach which we did was agriculturally relevant microbes, whether they could be pathogens or they could be symbionts, which could be helping the plants. So we were studying their metabolism very closely since last 5 years. Then this allowed us to, the progress has allowed us to right now combine both these parallel streams and enter into the field of plant microbial metabolic crosstalk, which happens to be much more challenging, as already Kushboo has presented some impending challenges and how how some progress can be made in this area using stable isotopes. So my expertise with flux analysis under steady state conditions, followed by in both plant as well as microbes, we think we could make some contribution in plant microbial metabolic crosstalk, but we ended up with more questions and challenges, as you will see in the talk. Apart from that, there are few projects, apart from the fundamental questions we are asking, few projects which we are undertaking in the interest of the global agricultural scenario, especially to the food security, that is like Farmer Zone, the future of agriculture in the smart farming, and other applicable projects like, say, understanding the diversity of the phytochemicals, which are revenue sources for the Himalayan population, and also converting agro waste to valuables, where a couple of my scholars are very keen on it. Having said that, these all are funded by various funding sources, for which I really acknowledge them. And over the few years, we established various prototypes and facilities, which I feel like could help, which we feel like sharing with others. Anyone who wanted to very closely work with our group, please feel free to get in touch with us. We can give you access to GCMS. I think that is the main workhorse, and other plant culture labs, CO2 controlled plant chamber established by Maneesh. And also we do have some smart farm, or you can say a smart farming application, Farmer Zone app, which is available in the Play Store. You can download, and potato farmers can really get benefited out of it. And one important aspect while we developed this app is that several farmers are very keen on understanding the disease detection or disease diagnosis in potato plants, in which blight is one of the diseases. So that leads me to the fundamental questions again, which we all have been asking in our labs, that is about understanding phytopathogens or symbionts, which are directly relevant to our crop productivity and food security. So why should we really care about these phytopathogens in the first place? If you look at it, these are relevant to the food security and they lead to the crop loss. For example, here, Ralstonia solanacearum, which I work with, causes wilt disease, and Xanthomonas, which Xanthomonas various species are there, which causes like blight by Xanthomonas oryzae and Xanthomonas campestris causing black rot disease. Now, these are among the top 10 bacterial phytopathogens, and their metabolism is poorly understood. So that is how I diversified after my PhD and postdoc to enter into the phytopathogen metabolism, because their metabolism was poorly understood. So in order to better get insights into this, we have to establish the crosstalk between the plant and microbe. There are various questions here which one would need to really understand or wanted to address. Whether can we really mimic the plant host and see how the microbe behave under it? Do we need to integrate several omics together to come out with a model which could be very which could be predictive and help us in readily controlling them, various strategies? Now, what are the key pathways and fluxes? Do we need to come out with novel approaches? In fact, our study shows that we need to come out with novel approaches of stabilized isotope analysis, and several physiological questions like, say, whether there are cellular demands, whether there are any marker metabolites, what metabolites are really exchanging, and any cues to control these pathogens. As Poonam has just presented, ED pathway could be inhibited to control the wilt disease, if at all we could find potential molecules which could do them in a more natural condition. So these two pathogens which my lab has been focusing on, the Ralstonia solanacearum and Xanthomonas species, they cause havoc, in fact, affecting our food security globally, both via the wilt disease and the blight and rot, and major plant crop species. So we should care for them, of course. So what is our approach for that? The approach which we undertook is that we would feed these organisms with multiple single or multiple carbon or nitrogen sources, mainly in the free or mimicking condition, and then undertake a systems approach wherein we would have transcriptome data along with the 13C MFA data and genome scale reconstruction based on the genome sequences available, wherein we can bring in the flux balance analysis of the day one, all the skills. And then along with that, the metabolome profiling data under various conditions. So using all that, we wanted to come out with a predictive modeling framework at the end, so that which will incorporate all these various strategies into a single integrative network. So that we believe will delineate the poorly understood metabolic phenotypes of these phytopathogens. So here I didn't touch upon the in-planta aspect, which will be the focus of this particular talk. As we go across, I will speak about that further. So just now Poonam has explained how she managed to get the flux map of Ralstonia solanacearum using parallel 13C labeling strategy. Parallel because that would give us more confidence in relation to the error bars on the fluxes. So she precisely could decipher the phytopathogens under minimal media. Also, which she also did work on xylem mimicking media condition, which is mimicked to be the xylem composition of the tomato host, wherein glucose and glutamine both were fed to the system, and then we managed to get the precise metabolic phenotype. But that doesn't explain anything about the in-planta yet. Are we near or are we far from the reality? So that brings us to the in-planta, so that we can really understand the metabolic crosstalk, and we end up actually it to be more challenging. And as you can see here, there are several challenging limitations. Let me just turn on my laser point. Some of the limitations is the cellular heterogeneity. The plant cells plus the bacterial. Again, within plant cells, there are different cell types. So the cellular heterogeneity would greatly influence. And conventional MFA does not necessarily yield averaged flux values as well. So we do have these challenges, compounded further by the day-night cycles in the host, and virulence versus metabolism, which are as a trade-off by these phytopathogens, has to be considered as well. And there are several temporal and spatial challenges. So here, with these many challenges, we figured out that doing metabolomics or other studies would give us whether there are any differences or not, but it is not going to define the precise metabolic phenotypes. So that led us to further look into what kind of solutions we can adopt. Then comes again George and Nick's. I should acknowledge Maria here, who have published and has demonstrated that cell type specific metabolic flux analysis can be mainly based on reporter proteins, can be actually used to capture targeted cell fluxes or its metabolism. So clearly it has been shown that cell type specific GFP expression, green fluorescent protein expression, could be a potential source for protein based amino acids which can report on the central metabolic fluxes. So that we picked up, and that is when I got back to my roots again. That's I connected with George and Nick in relation to shaping it up into a Spark project, which is one of the outcome is this workshop. So before adopting that technology, that is cell type specific tech using the GFP as a marker protein, I wanted to first of all explore if Ralstonia solanacearum colonizes the xylem properly. That has to be demonstrated. Excellent work by Kathleen Allen, again from the University of Wisconsin, Madison, has clearly shown that GFP tagged R solanacearum in mid-stem can clearly be seen to be growing in the xylem vessels. Both in the electron microscopy also shows that under infection, all these Ralstonia solanacearum harbors in the xylem vessels, thereby infecting, clogging actually the water flow or the xylem flow, and then the plant eventually wilts out. So we wanted to adopt this technique of the GFP tag, and readily when on my search within India, I figured out Professor Suvendra Ray's group at Tejpur University has got this M. cherry strain, which M. cherry strain of Ralstonia solanacearum, and they have also not only that, they also have demonstrated that there are two ways of getting this Ralstonia solanacearum into the plant seedlings, either via root dip method or via the leaf cut method. There are two important methods which they established, and the references provided below can be a good source where you can connect it and further look into further details. And here clearly they have shown that in fact whether it is leaf, stem, or root, the red M. cherry could be seen in the xylem vessels. Even here you can see that on the infected seedlings. So that gave us the confidence that to see if we can adopt it as a cell type specific for capturing cell type specific labeling information of the Ralstonia within the plant, and see whether we can tap into any metabolic crosstalk happening between these phytopathogen and the plant. So this is the question which we wanted to ask. So the question is whether the cell type specific 13C MFA method is really feasible in Ralstonia solanacearum or not. So we quickly adopted Poonam's methodologies, where we fed 1,13C, 1,2,13C glucose and universally labeled glucose to Ralstonia solanacearum, which has got the reporter protein of M. cherry. And then we compared the total cell protein versus the reporter protein, I mean the complete hydrolysate with M. cherry, and did 13C MFA to see if there is any difference between, if there is any influence of reporter protein on the central metabolic fluxes. So that we have to do to validate that this is indeed a proxy for the complete metabolism, capturing the complete metabolic flux analysis. The right, you can see the mass isotopomer distributions of the F1C1 versus F1C1 M. cherry protein hydrolysates. All these amino acids have clearly shown that, in fact, both of them can be represent the same flux map which Poonam has presented under the minimal media condition. So again, the ED pathway, just to summarize what Poonam has really found, in both the conditions, F1C1 and F1C1 mcherry. And you can also see that the phytopathogen actually, the anapleuritic reactions as well as the TCA cycle, most of the fluxes can precisely be defined with very less error bars, which means that they would be more confident in relation to the flux estimates. All the biomass estimations and others were done in a very sophisticated and well controlled experiments by Poonam and other scholars. So that gave us the confidence that in fact mcherry expression doesn't influence the metabolism of this microbe per se, or the fluxes of this phytopathogen under free living condition. So now now the next obvious question for us is whether we can purify the mcherry, and then whether that particular purified mcherry based amino acids which are labeled can provide us the retrobiosynthetically report on the central metabolism or not. For that, we have to really purify this particular protein and then run the GCMS after the hydrolysis of that. And earlier work from George and Nick has shown that GCMS could be one of the factors here, or the sensitivity of the equipment because very less amount of protein would generally be available. Thankfully, we have a GCMS system which has got tenfold a bit higher sensitivity than the normal GCMS. That's what at least the Agilent makers have made us believe, and it is indeed so. We tested it, and here you can see the protocol we established. Or in initial attempts, we found that there are very non-specific bands. And so Poonam has further improvised this particular methodology, and then which led us to get purified mcherry proteins. And this mcherry proteins on purification, after we received it, after they were isolated, we hydrolyzed it further, and we got the amino acid fragments which would report onto the central metabolism of this Ralstonia solanacearum. So on doing flux analysis of those, that is mcherry F1C1 total cell protein versus mcherry F1C1 mcherry reporter protein. So here there is one takeaway. Mcherry reporter protein per se will not give you all the 20 amino acids. In fact, all the 15 amino acids which are possible to be measured using the GCMS on protein hydrolysis, some gets oxidized. But there are a few, I think about 10 to 11 amino acids can be reliably be measured in the mcherry. So on using the same amino acids in the total cell protein of mcherry versus mcherry reporter protein, which are isolated, we found that the FluxMAPS more or less agree among themselves. Again, ED pathway and all the other features which we earlier have seen in the Ralstonia solanacearum could reliably be captured. So that clearly shows that mcherry can be used as a proxy to define the central metabolism of Ralstonia solanacearum. Now, that is under the free living condition. So in planta, whether this is really possible or not. So this is work in progress, which I'll be sharing upfront what we have observed till now, and hopefully that will be useful and will be open to receive any suggestions further on this as well. So we came out with a strategy which we thought we should be adopting, called as the reciprocal 13C feeding to tomato seedlings. Either we feed the 13C to the microbe and then infect it to the plant, or we feed the plant first of all with 13CO2 or 13C glucose under different conditions. For example, let us say in one condition we feed 13C glucose, and then it can go to autotrophic condition, or it can go to the heterotrophic condition, and then the 13C tracking of the label could be done. And on infection, we can see what happens to the reporter protein by hydrolysis, whether the label entered into the mcherry amino acids or not. Sounds complex, but in fact, we some of our experiments have shown that it is possible. It is possible to adopt this technology. Now, in the second case, wherein on 13CO2 feeding, which might be very complex given the instationary conditions which needs to be adopted in such a short time may not actually label the M. cherry at all, or the Ralstonia solanacearum at all in the xylem. So in that case, we thought like we have to feed for at least 3 days and then track in the protein how the labeling is getting incorporated, at least in the light phase period. So here we are sure that there are a lot more questions here, whether where we may need to adopt only kinetic methodology for short time during the whole light phase, or maybe in the dark phase as well. So these are this is what we feel like will give us ultimately the metabolic crosstalk between the host and the pathogen in this case. So some preliminary experimental outcomes here, whether tomato seedlings themselves take glucose or not. In fact, there does. And the work has shown that under control and treatment, which means under infection, there will be differences in the metabolite profiles. We haven't shown any principal component analysis and so on, but some work has been done on that. And we have clearly seen that there are variations among that. And about 62 metabolites we captured, and we have shown that the glucose is even taken up by the system even under the photosynthetic condition. Now that doesn't guarantee that the 13C would still be getting into the M. cherry. So that work is still in progress, and we'd like to see if the 13C could be measured in various intermediates of the tomato seedlings. So 13C feeding to tomato plants. Two approaches we did. One is autotrophic condition, and another is autotrophic and heterotrophic condition. This is again done by Maneesh, who has presented the 13CO2 feeding experiments which he has done. So on doing that experiment, we figured out that if you do 13C in particular, we see label incorporation to same extent, whether it is heterotrophic or autotrophic condition. This is alanine 232 fragment, and we could see that there is about 5% average 13C label. Labeling in alanine 232. Similarly, in case of serine, which again is almost about 3 to 4% in between those. But there are some variations in relation to control and infection. So that would be of some interest in order to derive whether there is any differences in the 13C redistribution and control and infection conditions. What happens to these plants over the 72 hours is that they start wilting. Some of the wilted plants are quenched, and their metabolites were captured, and this analysis is a part of that. And we have taken the protein hydrolysate overall of the seedlings which are infected. That means these are a combination of plant cell plus the microbial cells in this case. We are still far from really isolating M. cherry from the seedlings in order to get clear picture about what is happening to the label redistribution in the Ralstonia solanacearum in the plants. So the further analysis of other amino acids, like say aspartate, which is coming from the node of the TCA cycle mainly. The aspartate as well, we have seen that they have got the labeling. That means even TCA cycle is labeled under the 13C6 control experiment and the infection experiment. Similar is the case here. Here are some of the experimental setup which Maneesh has done. And on close observation of all the other metabolites, what have been detected under the control and the infected, we observed that actually 3-hydroxybutyric acid, one is which is the precursor for the PHB, which Poonam has mentioned. We found that under the infection condition, it has enhanced its levels. Probably that could be acting as a marker metabolite. In case we don't get the sufficient amount of M. cherry, we could further use the 3-hydroxybutyric acid labeling in order to prove that whether the ED pathway is still active under the in-planta conditions or not. So these are some of the impending questions. And there are several other highlighted ones which Maneesh feels like could be potential marker metabolites which we need further focused attention to. Now, kinetic 13CO2 feeding under again control and infected has given us some preliminary leads, which shows that there could be differences between the control and the infected conditions. For example, here in alanine 260, one of the fragments, this I believe is from the, this is from the proteinogenic. That means the protein hydrolysate leading to the alanine, valine, and all the other amino acids. And we see there are differences on day 1, day 2, and day 3 in the incorporation of 13CO2 between the control and the infected. Similar is the case in some of the other amino acids. So this data needs to be further carefully evaluated. This is unpublished. Just I would state at this point, I wouldn't read too much into this, but this still, nevertheless, these point to the feasibility of adopting this particular methodology of feeding 13CO2 for longer durations, followed by protein hydrolysate, might have some valuable information which needs to be carefully tapped into to capture the plant microbial metabolic crosstalk. So another question which these outcomes have led to from the soluble metabolite analysis is that does Ralstonia solanacearum exploits host sucrose as a carbon source during xylem infection? So inside the xylem, we figured out that under infected conditions, the sucrose levels are reduced by several fold. Whether it is in the sucrose or whether it is in the shoot or in the root, we found that the infected ones have got depleted amounts of sucrose. And not only that, again, the labeling also reflects that there is some variations between the control and the infected. So more focused attention needs to be done, needs to be made to really understand what is happening in relation to the carbon utilization by the pathogen. Let's get into the one more interesting observation, which again made by Maneesh, is that under the infected condition, he figured out that the GABA, in fact, the GABA, I think this is the GABA, yeah, under the infected condition, the GABA has been completely depleted. Maybe that is being utilized by the pathogen. So this is again another question. And GABA and glutamate are coming from the nodes of the TCA cycle, wherein even the GS GOGAT cycles and the nitrogen would also play, nitrogen metabolism would also play a significant role. And here we are seeing that there are differences between the control and infected in terms of the average 13C incorporations from the 13CO2 experiment. So these gave us more questions rather than any answers at this point. I would share, and we are further looking into these data sets to make sense of all these findings. So I would like to give some highlights here. There are several potential metabolic signatures which could actually tap Ralstonia solanacearum's metabolism in planta, which is causing wilt disease. And some of them are like, say, sucrose, GABA, some of the other amino acids, which might play a significant role. We do have indications for those at this moment. We don't have any particular metabolic network-based crosstalk, but we look carefully into day 1, day 2, day 3 data sets, and probably we can derive something. And number 2, we can use the mCherry as has earlier been demonstrated, and we will be tapping into that particular aspect to capture exact 13C labeling in the mCherry amino acids in planta conditions kinetically. So that should give us some indications about what might be happening to the central metabolism in case of Ralstonia solanacearum in planta. So I would like to summarize with this much of all the background and what we have found. Cell type specific reporter proteins like, say, GFP, mCherry can indeed be adopted for host microbe metabolic crosstalk. More work needs to be done. Definitely it is in the positive direction. That's what I believe. We have more challenges to address, and as I shared, this is the work in progress. Reciprocal 13C feeding of host microbe can be the way forward. I would like to receive your comments on that. Any inputs would be highly appreciated. And 13CO2 studies can be indeed be very useful, because this is getting more near to the natural conditions, where we can integrate this along with other FBA-based dial cycle models and see whether we can validate all those model findings using 13CO2 studies. So with that, I would like to acknowledge again George and Nick mainly for readily agreeing to be on the Spark project and reinvigorating all our energies into it, and all the support. I think since last 1 year, in spite of the COVID, we had almost every month, sometimes twice a month, where we continuously discussed about this project, and Maneesh and Poonam, who have been executing, and you can see we do have some leads at this point, which needs further focused look into. I would also like to thank Biox Center, AMRC of IIT Mandi for all the facilities. Professor Suvendra Ray and Siddharth Satpathy, who have been on the Spark project as well, and giving us all the valuable insights in relation to the M-Cherry strain F1C1. Without those strains, we couldn't actually test some of our hypotheses, and also the funding source, which I've been receiving since last 5 years at IIT Mandi. And finally, my team at IIT Mandi, I really thank each one of them. In fact, this is all their hard work, which has resulted into progress, and it's a great pleasure working with every one of them. Thank you. I'll be happy to take any questions.
Thank you very much, Shyam. It's very impressive to see what you've achieved in the last 5-6 years since you started at IIT Mandi. It's also impressive that you're tackling such a challenging problem in looking at the plant microbe interactions. Let's see what comes up in the chat now by way of questions. Nothing as yet. Oh, here we go. Right, so this is from yes, from Wadzani Dada, I think it is. Yeah, it's been a brilliant and excellent concept. What was the basis for suggesting the potential biomarkers? And it goes on, since Ralstonia establishes a systemic infection, do these current studies see a possibility of exploring other plant tissues during the pathogenesis? So a couple of questions there. You can probably see them yourself in the chat because they came to all panelists.
Okay, I'll just try to. No, I can't see any questions, but yeah, is the question related to the marker metabolites, how they would be useful?
Well, yes, the first question is for suggesting the potential biomarkers.
Yes, in this case, say, for example, if we do have PHB, which is produced categorically by the pathogen, then we can use that as a potential biomarker, definitely. Whereas there could be some biomarkers which would be very plant specific, so those could also be used in this particular case. If you change it to other pathogens or maybe symbionts, let us say other pathogens which have got very specific metabolites, like say even there is this exopolysaccharide. If you do have that, which some microorganisms produce via the quorum sensing mechanism, if you can tap into the polymer and then hydrolyze them, maybe those molecules can also be a potential biomarker. So what we are trying to achieve here is that the 13C first of all enters into the host. Now the 13C host is ready. The moment the pathogen goes and tries to infect, before infection, it will there is a trade-off between metabolism and pathogenesis. So what the pathogen would try to do is first of all divide itself slowly, multiply it, travel to different parts of the plant, and then it starts consuming the carbon resources which are available in the plant. So if this 13C are already available, now the pathogen will multiply. So definitely there would be new protein which it would be producing. So we believe this M-Cherry or maybe GFP protein, which would be labeled, will have specific information of the pathogen metabolism. Because on hydrolysis, we are looking back into the amino acids which are emanating from the central metabolism of the phytopathogen in this case. So the same concept can be adopted to other pathogens for sure.
Yeah, the second part of that question referred to the systemic nature of the infection. and the question was wondering whether you'd be exploring the impact on other plant tissues during pathogenesis.
In fact, this Ralstonia solanacearum infects xylem, as we have clearly seen. So, in case of plant tissues, I'm not sure at this point whether we can go ahead with like root, shoot versus leaf, whether there would be any specific differences among them. But we adopted seedlings to begin with, and once we validate if there is any merits of doing that, we would be doing it. For example, say if we wanted to study nitrogen fixation by rhizobium, say another model system. In that case, we would be more specifically should be looking at the root, as George has yesterday presented, that within that nodule as well, there are different layers of activities where the nitrogen fixing would be in a particular zone. So yeah, but that is the beauty of cell type specific marker proteins. Probably we could tap into that, which would be expressed in a specific specific tissue of the plant, and then tap that. Why not? We may have a GFP plant with specific marker protein and M. cherry from the pathogen or the symbiont. Yeah, maybe that's a good suggestion.
And then there's a question from Bertrand Gaquier, who comments firstly, very interesting talk. Do you plan to compare sensitive versus resistant tomato cultivars where Ralstonia doesn't colonize the central lumen despite being present, to make a new metabolic comparison of the interaction?
Oh, thank you. And in fact, this is a very interesting question. On discussions with our collaborators, Professor Ray from Tejpur, he did mention about this particular aspect, where he says that although several tomato cultivars are resistant or some are susceptible, within the susceptible as well, there are few escapees. That means on infection as well, at least 10% of the plants try to escape. And his question was, how are they escape the effect of the pathogen? I mean, they doesn't wilt. So his question was, number one, within susceptible, can you discriminate what is happening in terms of metabolic interaction? And once they already, if there are resistant varieties, can you again discriminate between them? I think that would be the future direction. As of now, via metabolomics, we are attempting to see what kind of differences do they have between resistant and susceptible varieties. Some literature is also available in that, but 13C none. Yeah.
Thank you. Any more questions? I don't see any more in the chat at the moment. We'll just wait a minute. No, it looks like that may have exhausted the questions, Shyam. So I think at this point, I hand it back to you. And doing so, thank everyone who's spoken in this afternoon session. 3 very interesting talks. Well, actually 4 talks, because Kushboo gave us 2 talks on different topics, which was very appropriate, very good. So over to you.
Thank you, George, for the whole session and all the other speakers as well. So that ends up all the talks, day 1 and day 2. Thanks to all the participants as well as the speakers, panelists. So before we close, I first of all request Professor Nick Kruger to kindly summarize today's day 2 sessions, and what are the major takeaways which we should be taking from that. Yeah, Nick, please, if you don't mind.
Yeah. Okay, yeah, happy to do that. So thank you, Shyam, for the invitation to make some concluding comments, if only to ensure that I paid attention for the whole of today and all of the fascinating talks that we've had on metabolic flux, on metabolic flux analysis. What strikes me about the talks we've heard in the 2 sessions today is how well they reflect current state of the art in plant metabolic flux analysis. And So in commenting on the lessons we've learned in these sessions, I think it's essentially asking me to assess the status of MFA in plants. And that's difficult for me to do, except through the prism of my own concerns and prejudices. The first thing, though, that strikes me from the talks we've heard is the diversity of biological problems that are being addressed in plants and their associated microorganisms, be they pathogens or beneficial microbes. I'm also struck by the fact that almost all of the studies that we've heard about in today's sessions have employed steady-state flux analysis. And as I suggested in my opening outline, these analyses have all required very careful refinement of what would be called the standard process. They're also largely confined to plant cell cultures, and plant cell cultures under a restricted range of conditions that remain amenable to analysis, and particularly to the constraints of achieving a metabolic steady state, and under most circumstances achieving an isotopic steady state as well. Of course, there have been exceptions to this. Ed, in particular, talked about his work on non-stationary flux analysis. And I think there's an important need for us to move away from steady-state flux analysis, where possible, towards non-stationary flux analysis, if only to increase the range of questions that we apply our flux analyses to. Ultimately, of course, we would also want to be able to apply these strategies to systems in which there are dynamic changes in metabolism. Of course, you could say that these problems disappear if your focus is microorganisms rather than plants, because they can be grown under defined cultured conditions. But as we've seen again in several talks, very often the interesting and important questions to ask about plant-related microorganisms is not necessarily how they behave in isolation, but how they behave in interactions with plants. And those interactions necessarily involve, one, heterogeneous systems, so compartmentation and how we can dissect the metabolic events in different parts of the association become important. And secondly, those interactions are by their very nature also dynamic. They're not steady state. So I don't think we can escape the challenges of trying to refine metabolic flux analysis into a more dynamic system simply by avoiding plants and focusing on microorganisms. That's not going to work. We have to move towards a mechanism of being able to move towards dynamic flux analysis, and dynamic flux analysis probably in whole plants. Indeed, as emphasized by a comment that was made by Barclay Walker in the chat line, he raised the exciting possibility of not simply studying plants in the laboratory, but moving the entire system into the field and natural growing conditions. And whilst that's technically extremely challenging at the moment,